So for our first talk, we'll have our CEO, Dr. Baozhen Shan, or what more of you are familiar with. His name is Paul, and he's going to be talking about an enhanced workflow for DIA data analysis. Now, Paul began working at BSI in 2008. He has played a key role in developing the company's main products, Peak Software. And he obtained his first PhD in 1997 in chemistry from Nanjing University, and his second PhD in 2009 in computer science from the University of Western Ontario. Since his leadership at BSI, Dr. Shan has integrated mass spec services among other protein characterization solutions to the company as well. And so, Paul, whenever you're ready.
Thanks, Cassidy, for the introduction, and thanks all for joining our yearly user meeting. And I take this opportunity to share one of the features we put in our online product we call online DIA. It's a deep learning-based solution to DIA data analysis with a streamlined workflow for peptide identification. So my talk mainly have three parts. I first introduce the deep learning tools used in Peaks Online for DI data analysis, followed by a streamlined workflow to enhance the peptide identification, which contains spectral library search, direct database search, and de novo sequencing. Last, I use some case to show the features of this streamlined workflow. And deep learning has been used in proteomics recent years, and it brings some advantages and bring the proteomics to some deep level. In Peaks Online, we used many deep learning knowledge tools for DI data analysis. Specifically, we know that retain time and the CCS value is very important for peptide identification. And different deep learning model was used for retain time prediction and CCS value prediction. The typical work here on list is in 2018, a deep RT was reported, and AISTM and CNN are used to predict the retain time given a peptide sequence. Surprisingly, the correlation between predicted retain time and observed retain time reached to the 0.99 the correlation value. That's bring the identification to the next level. Also, the similar deep learning model are used for CCS value prediction from timsTOF data, and it make the CCS value predicted one and observed one within 1% variance. Combine those retain time and the CCS value with the spectra fragment area can significantly improve the identification. Also, the precursor feature prediction and detection has been improved by deep learning technology. Typical work you can see reported in this year is about given a database and an enzyme, we can predict the peptide detectability and in the mass spectrometry experiment. And also we can use deep learning technology to extract the precursor feature abundance. With those two, we can increase or reduce the false positive or increase the accuracy of quantification. And meantime, we also know this one. And given a peptide sequence and with deep learning technology, like here is the one they use bidirectional AISTM and with CNN and was used to predict spectra. And this work is pioneered and bring the spectral prediction to a fantastic level, and it makes the predicted spectra and real spectra the correlation value more than 95%, and that make a database search really fast and sensitive for DIA data analysis. Traditionally, we know that for database search, and we given a database, protein sequence database, we use enzyme to generate all possible peptide. For each peptide, we have a theoretical spectra, and then we compare spectra with real spectra. We assign a score. Finally, we report the identification result with false discovery rate estimation. With similar ideas, so we can use the spectral prediction for database search of DIA, and without creating a spectral library with the same project with DDA, and this we call direct database search. The pioneer work you can list here is reported last year, and by different groups. So on the other hand, de novo sequencing try to answer the question in the opposite way. In data spectral prediction, is given a spectra, we try to see, given us peptide, we try to see this peptide generate what kind of spectra. And in de novo sequencing, it's just opposite way. Given a spectra, we try to see what are those peptides generate those spectra. And so we, in our group, we started using CNN and AIST and to extract the spectral information features and to mimic the spectra and the chromatogram information. And we reported this result in 2017. And later on, we extended those de novo sequencing from DDA to DIA by incorporating the chromatogram information of precursor and given to get de novo sequencing result from DIA data. In PICS Online, we improved all those technologies for spectral prediction, precursor prediction, retain time prediction, and de novo sequencing. We put those tools integrate in PICS Online for DI data analysis. More than that, we provide a streamlined workflow for peptide identification. Specifically, given a data, we have 3 strategies to explain, to annotate the spectra. There are library search, try to find some peptide which we already know, and or a database, we try to find a segment of those protein sequence which we believe generate those spectra. Or we can use de novo sequencing, and to directly from the spectra we get the peptide sequence. We put them in this way. Given a data, we first check if there are any peptides in the spectral library, and to have the evidence in the data, and we use accurate FDR to get those control, to get those peptides from the library. And then for remaining unidentified precursor, we see if there are any evidence from this protein sequence database to show the confidence in the data in this data, so that we can get database peptide from the data. Last, for those remaining unidentified precursors, then we follow by de novo sequencing to get de novo peptide. Normally, those peptides are from unexpected processing by proteomics, or from long coding region, or from some unknown out of frame, within frame, like synthesis. And not just put them together in a pipeline, we also take the advantage of the known peptide confident one to extend it to those larger space, control the FDR. For example, the peptide that we get from the spectral library, those are most common confident peptide because they have experimental evidence from our traditional previous experiment. Those are very often very confident. Then we use transfer learning technology to extract this feature specific for this data sets to pass those information to the following deep learning technology to get the peptide identification more fit in this data, so that we can get higher identification rate and lower false discovery rate. So this is a workflow we put in online for DI data analysis. How it works. And first, we take a look at individual module for each function. Here is the spectral library search. We use bidirectional search. What the bidirectional means, given a precursor, we first check if this precursor, peptide precursor in the data have any evidence. Also, meantime, we use feature detection, we use prediction. We see whether if given a precursor in the data, whether there are corresponding parts in the spectral library. By two-directional search, we can significantly improve the identification rate and also control the false discovery rate. And here I list two benchmark with common data sets. The first one is relatively simple, is AB from ABIF PRG study group in 2018 for DIA study. And the data was generated from High-Throughput Digest. And there is 3 DDA round to generate library and 3 DIA round to see their identification rate and their reproducibility. We benchmarked with spectral large, DINN, and to see how it works. From this one, you can see on the middle, in the middle, PICS online and the DIN gives very similar identification rate and slightly better than like a spectral lot. Surprisingly, the overlapping between different rounds shows very high reproducibility. For PICS, even reach like a 96% overlapping. And that really shows that like a DIA unbiased in like a unique feature for proteomics. And following this one, we test the library search with a little more complicated data sets, which just published this year. It's from like for Immunopeptidomics. They with this data sets, you can see and PICS and the deep learning technology with all those integrate solution make some difference. And you can see PICS find more 7 to 8% more peptides compared to DIN and also showed a very good overlapping and more than 95% reproducibility among those replicates around. Okay, and our like spectral library search support ion mobility dimension. That means what people call the 4D spectral library search. The ion mobility will bring some unique power to resolve those isobaric peptides. And the second one is direct database search. During our research, we've encountered two major challenges. One is searching time, another one is sensitivity. The search time, because normally for each proteomic database and given an enzyme, they have a huge number of all possible peptide feature which could be observed in the data. And if they use deep learning technology to predict, you will have a huge pseudo spectral library. And there we are bringing two issues. One is non-search time. Another one is higher noise and highest false positive rate. So to resolve this, we extend our bidirectional search to direct database search. And surprisingly, it also gave good result. We benchmarked the data, and so that with like above data in recent publication. And here I show the result compared to DINN and PICS direct database search semi-triptych search. And but DINN at this moment they only support the full triptych search. So and we restrict the PICS use the same like triptych search, and you can see the overlapping is very good, and they are PICS and the DIN get very similar result. And we also have like for DI we can do de novo sequencing, and there for each detected precursor we find corresponding fragment ion spectra which have similar elution time. And for de novo sequencing, the main issue is to resolve pick reuse, because some fragment ion may shared by two different sequences, and this is a challenge for getting higher accuracy and lower false positive result in de novo sequencing. And here we showed some of those results, and we from the deep learning technology implemented in PICS Online. And that if we combine the three of those into a streamlined workflow, here I list some data here that you experience the advantage. You can see, given the data sets, which is a HeLa digest around 160 minutes in the lumos, and we have 3 DDA to generate libraries, 3 DI. With the DI data, you can see, we only need 18 minutes to get finished spectral library search from loading data to the result. If we get data library and the direct database search for those data, only take less than 1 hour, including de novo sequencing, and take 1.5 hours, which is much less than machine instrument time. And so that you can see those integrate workflow and not only bring the identification or to very high identification rate, also ensure that running time is within the machine time. And from the data, you can see this workflow can increase the double or even more than double like identification rate compared just to use some project or just to use a single identification method. Not only like deep learning can help identification, also can help quantification to get higher accurate result. And we can use those one for information from MS1 or MS2 for label-free quantification. Here is our preliminary testing result with HYE data sets, human, yeast, and E. coli. We mix them in a common way, which are reported in many publications. And this data was generated by our application team. And and here is the initial test. From this, you can see that it does improve the accuracy and with relative sensitivity comparable to DINN, and both the number of quantified protein groups and the quantification accuracy. So overall, we also provide a very good user interface for validation about the peptide identification. Given that we show the matched peak in the DI data from fragment ion and precursor ion, they are correlation, and we also show that 2 ways giving us identification, what's the data in the data and what's the predicted one, so that we can give the full picture from deep learning technology perspective. And here is like a snapshot of the label-free quantification with those standard data sets, like a mixture, and to show they give really good quantification result. So, and in the last part, I try to use recent publication to show the power of those streamlined workflow in peptide identification. And recently, Immunopeptidomics and bring lots of challenge. People believe DI data might give a full image about the complexity of immunopeptidome. And this work try to leverage a different library from more than up to 20 projects to build a larger library and to increase the library search in sensitivity. Also try to use deep learning technology to improve the data analysis. And with this work, they are successfully found, compared to original one, just use traditional library search, increase almost 2 to 3 times compared to mass clone only result. And so that's really fascinating. We take this data sets to see if use our workflow, what we can get. So that we look at the library search. Here is the result. In the middle, you can see that we found much more peptides compared to the publication. On the bottom part, you can see the gray line is the reported numbers in the publication, and the yellow one, the dark green one is database. In the middle is spectral library search. And if we take a length line, select those one on the right is their motif deconvolution for the 6 years. And you can see our library search and the reported like a library search result has very, very similar motif. This shows that we are not only find more peptides, also the library search with good accuracy. And after this one, we can do library search and the database search together and get more peptides. Here I showed in the left part showed the peptides identified from the data sets. The small one is reported, and the middle one is library search, and the outside one is library followed by direct database search. And I also list here the motif the convolution result and compared to the reported. Again, you can see those one keep accuracy about compared to the spectral library only, and this also showed confirmed our observation. Direct database helped with deep learning does improve the false discovery rate. And more than those one, and we also found answer one question in the publication, because they are for this cell line, JY cell line, and they missed one expected motif, and the possible reason probably is have a lower number, and either the sensitivity is not enough. And with our pipeline, we are successfully find those one, and the motif is as expected. Indeed, the number of peptides is much less than other two years. And I think with this one, and you can get more peptide, and as we expected. The last one is de novo, the final step, and find more peptides. And surprisingly, those de novo only peptides and have very similar motif deconvolution compared to the one in the library, and they only show slightly difference, and this and also meet with similar observation or prediction in the many publication. And so with those one, I hope I can show some result to convince you or the pipeline does improve the peptide identification rate. How about DDA? Actually, deep learning also can help DDA. I won't have much time to talk about here. Just show one recent publication from Kust group. They use deep learning to boost the peptide identification. And they here I showed the one, if we just use max quantum to analyze the data, and the yellow one is, orange one is in the publication. The blue one is from online result. And with a similar observation is deep learning does help the peptide identification. And with that, we appreciate. I thank you for our correction team, especially for those research professors. They help give us some direction about our software algorithm development. Also, we really appreciate the team's efforts. We have an AI team in BSI. They constantly focus on to develop those algorithm and the models of deep learning to resolve proteomics data analysis challenge. Last, I thank you for your constant support to our research, and give us feedbacks, help us to develop good software to serve the community. Thank you for your time.
Great, thank you, Paul. There are a couple of questions here. Paul, I can read it out for you. One is, what was the nature of the de novo peptides that were added? There was a slide where the runtime is compared.
Oh, the running time only increased, like say, compatible to database search time. Yeah, you can see, yeah. Because in this table, and not for all precursors, we first do library search. After that one, as part of the precursor, we are not passed to like a database, and after database and the library, and the rest do the de novo. If we just do the de novo for our full data sets, it's take about 1 hour for all precursor for this data sets. Yes.
Okay, great. And then another question is, are there any plans of including Waters ion mobility data support within Peak Studio?
Yes, actually, we are keep watching, keep doing those one, and those, especially those technical ion mobility. I think that my we did some like a research. We saw some publication. They did bring some possible resolution power for those isobaric peptides.
Okay, and then last question here is, can data sets with no enzyme digestion and ETD or HCD fragmentation be analyzed with this workflow?
Yes, actually, and the data, like this data sets I showed here, and none of them, like say, current, like DINing, they don't do direct database search for non-enzyme data sets. To my knowledge, this workflow is the only workflow can do non-enzyme for direct database search.
Great. And then actually there's one more question. What is the accuracy of identifying peptides with PTMs such as citrullination?
And I think of now for DIA with deep learning method in DIA data analysis, still have some challenges for PDM analysis. Surely, if given for common modifications, we can train those one. And theoretically, with enough data sets, we can do whatever modification you want. But in reality, you need data sets for the training. And the good news is for common PDMs, including saturation and current sort model can handle those one. But when I say the change is you are not versatile like DDA data, you can do open PDM search. For DIA data, still have some changes there. We are working on those.
Perfect. Okay. Well, thank you, Paul, for that great talk.
You're welcome.
Great. And then Cristina, if you would like to go ahead and share your screen. So we're very excited to have Dr. Cristina C. Clement present her work here for us today. She has been a longtime user of Peaks, and I mean, I can even remember meeting her in my very first ASMS conference at the Peaks booth. And Dr. Clement graduated from the School of Biological Sciences at the University of Missouri, Kansas City in 1998 with a Master of Science in Molecular and Cellular Biology. She then continued on to the City University of New York, where she graduated with a PhD in biochemistry in 2006. And after that, Dr. Clement held two postdoctoral appointments, one in the Department of Molecular Pharmacology at the Sloan Kettering Institute, and the second in Pathology Department of Albert Einstein College of Medicine. Among her most important contributions to the research worth mentioning, the development of wide variety of nano LC-MS based assays, including targeted peptidomics, lipidomics, and proteomics for analysis of many biological fluids, and which led to the discovery of biomarkers for the pathophysiological conditions, including juvenile arthritis and other autoimmune diseases. In her current position of Assistant Professor of Research in the Radiation Oncology Department at Weill Cornell Medicine, Dr. Clement will continue to develop and apply promising new mass spectrometry, chemical biology, and molecular pharmacology technologies, aiming to reveal the molecular mechanisms that change the antigen processing and presentation of peptidomes and proteomes in dendritic cells under healthy and diseased states. So, Cristina, over to you.
Hi, Catherine. First of all, I would like to thank so much for the introduction. I am indeed a long-time user of PICS. I started to implement PICS in our labs by 2009, 2010. And since then, we were quite successfully to show how advantageous was to use such bioinformatic platforms, both for peptidomics and proteomics analysis. So today I'm gonna try to give you a different perspective on the immunopeptidomes, and I choose to show one of our last research, which focus on understanding the antigen processing in late endosomal compartment. This slide shows a disclosure, my position at Weill Cornell, and the main funding sources. And my computer. I'm sorry, but it looks like so I'm quite sorry. It looks like I have a problem to move my slide in the. Okay, so first of all, I would like to acknowledge the Englander Institute, Precision Medicine, and Department of Radiation Oncology, Weill Cornell, and then Columbia University for providing us with nano-LCL MSMS runs analysis of our samples, providing the raw files. Then I would like to thanks a lot to Dr. Lawrence Stern from University Medical School of Massachusetts and her postdocs, Dr. Nanwer, and then of course the bioinformatics solutions, Catherine, Dan, Ida, and Caspar for the long-time support in our research. Our lab is focusing primarily on understanding the antigen processing and presentation. In the left side of this screen, you can see a typical antigen presenting cells, which it is dendritic cells. So the antigens from extracellular compartments or from intracellular compartments are cleaved inside the late endosomal compartment in very small peptides, as some of you already know. There is an exchange with a clip peptide on the invariant chain, and that exchange further on, it is mediated by the chaperone DM. The peptides are loaded on the MAC class 2 and then transported on the cell surface, presented to MAC class 2, and thus they represent the immunopeptidome. Under inflammatory conditions, and that is reported by many research in the field, there are changes associated with the distribution of immunopeptidome. First of all, the MAC class 2 molecules increase in their expression on the cell surface. Then there is, of course, a differential type of exposure of the peptide epitopes. The low acidic pH under inflammatory condition in the late endosome could change actually the affinity of some peptide epitopes during the exchange with the clip, and the editing could be different. Moreover, there could be some additional post-translational modifications which can accumulate on both the antigen processing proteins like the cathepsins and also on the clip or on the DM. And altogether, this can have a significant effect on the final immunopeptidome, which you actually sequence, as you could see in the previous talk of Dr. Paul. There are many reports in the literature, many studies on the immunopeptidome displayed both on MSc class 1 and MSc class 2. So what is very important to know for this audience is that even you have a high resolution sequencing of the immunopeptidome, sometimes these peptides may not have any kind of immunological significance without engaging to the TCR presentation. And here I just remind you, typical immunological synapse where you see the CD4 T cells recognizing the MHC class 2 with the peptide in the complex. So during the inflammatory conditions, and I'm talking about redox type of changes in the microenvironment inside the cells, like in the type 2 diabetics, autoimmune disease, and cancer, we hypothesize that actually both the MHC class 2 synthesis, antigen processing, and peptide loading pathways will have dynamic changes, and that will be reflected in the final immunopeptidome, which is displayed on MHC class 2. So we are looking into understanding how are these redox-mediated proteomic and peptidomic changes affecting the efficiency of protein processing inside the late endosomal compartment. Then how is this in turn affecting the DM, editing, the selection, and the presentation on MHC class 2, and then in turn, how is that reflected on the immunological recognition by the TCR CD4 positive T cells. So we recently published this year two main key studies. First of all, we proved in immunity paper where we share with Dr. Lawrence Stern lab this type of authorship that post-translational modification which were accumulated on both MHC class 2 and intracellularly on some key proteins like DM in editing, and also the immunopeptidome, which was presented by MHC class 2 from diabetic type mice, like OB mice, were quite different than those presented by the MHC class 2 under steady state physiological conditions. So we have two types of mice which we are investigating, the control mice, C57 vaccines and OB. On the other side of the matter, we also proved the late endosomal compartment accumulate a lot of post-translational modification, and somehow they will have a different type of processing capability of the antigens, and also the peptidomes derived from this antigen could be different because of the different type of redox accumulated post-translational modifications. So just to introduce a little bit more the system, the two mice model, actually there are three. We have a control mice, and then the mice model for the metabolic oxidative stress is OB and high-fat, high fructose diet mouse. We wanted first to be sure that indeed this is a good biological system to investigate. So we actually prove at the level of different organ and then tissues that we have increased oxidative modification, as you can see here, both in the OB and the high-fat fructose diet mouse, and almost all the main organs from the spleen, like secondary lymph organs, and then kidney, heart, fat tissue were systematically accumulating the redox stress conditions. We prove also that the CD11C, the dendritic cells, are our main cells we are focusing on, are increasing the inflammatory conditions. So in addition, we use primary mouse dendritic cells from the splenocytes of these mice models, and we prove the advanced glycation and products and the carbonylation were enhancing the proteomes of these cells. So overall speaking, in this moment, we were able to consider that these mice models were suitable for our interest to analyze the immunopeptidome and the latent endosomal compartment processing of the antigen under stress, oxidative stress conditions. Next, also we further on have the sera of the mice metabolic profiling, and we were able also to convince ourselves that the glucose, insulin, cholesterol, or also increasing the OB than in the control mice. So our working model proposed that qualitative and quantitative changes in the proteomic landscape of the dendritic cells, together with the accumulation of post-translational modification, and I'm showing here as example two main, the methyl glyoxal ADAX and 3-deoxyglucosan on arginine. All this together would affect the antigen processing, and further down the line will have qualitative and quantitative effects on the immunopeptidome display on MAC class 2. So the main question we wanted to respond, and we were interested to figure out, is that if you have these proteomics changes and accumulation of PTM during the metabolic redox stress in the proteome, how is that reflected in the end at the level of immunopeptidome? And of course, we wanted to even more understand the latent endosomal processing of antigens, which could indeed affect the immunopeptidome display, the affinity, and then the diversity on MAC class 2. So the main aims and experimental design was first focusing on proteome and the immunopeptidome, and secondly on antigen processing, which use a latent endosomal organelle, which were purified from control mice, dendritic cells, and from the OB or high fructose diet, fat diet mice. So we use a typical orbital fusion trimass spectrometer, and in some cases few exactly, to Dr. Gessoni from Irving Cancer Center in Columbia. And we use label-free DIA, DDA, combined methods. We generated our own spectral libraries from the proteome and peptidomes of raw control and OB mice. And we were able then to perform labor-free quantitative analysis, identifying changes in different antigen or peptides, as I'm going to show in the next section of the talk. So before I'm going further on, I just want to show that the proteomic changes were quite significant in the high-fat, high-fructose diet or OB as compared with control mice. Here I'm just showing a very fast EPA readout in which quantitative analysis shows the oxidative metabolism proteome from different pathways involving TCA cycle, amino acid degradation, glycolysis, glucogenesis, and metabolic stress response, all of them upregulated in OB, HF mice as compared with control, and differential effect on the mitochondrial respiratory chain, in addition to an increase in the inflammatory cytokines. Remarkably, the STAT pathway was upregulated as the level of the proteome in the high-fat, high-fructose and OB mice as compared with control. So today there were a lot of questions related with post-translational modification. We always use peaks to identify the high-resolution post-translational modification. Here you can see which types. We are looking on the shift-based type of PTM, which are mediated by carbonylated moieties from oxidized sugar like methyl glyoxide, with the amino group of different proteins. These shift-based transform in Amadori ADAX and they get reduced. So we have all these varieties in PTM which are identified in the proteome of the OB high-fat, high-fructose mice as compared with control. They are increased. We do actually use peaks at the level of MS1 analysis and MS2 analysis to identify with high accuracy. You can see here an arginine modifying cathepsinase, which is one of the enzymes which process the antigens. And we have side-by-side the spectra of modified and unmodified. So we have a high confidence in using peaks to identify this redox-mediated post-translational modification, both in the proteome and the immunopeptidomes. We use a typical fast passable for the proteomics analysis in the late endosomal compartment and of the total proteome from the dendritic cells. And we just show here a typical readout where we were able to identify a multitude of PTM derived from the oxidized sugar moieties reacting with amino groups of different proteins. So this is a typical readout in peaks for the type of post-translational modification we were able to map to the proteome, as I said, of the total proteome of the dendritic cells and the proteome of purified late endosomal compartments from control OB or high-fat, high-fructose diet mouse. And as Dr. Paul mentioned earlier, we are using both DDA and DIA just in the immunopeptidome. I'm just showing here how the DIA data were deconvoluted by a search here, spectral independent, library independent. And in the next slide, you can see here spectral both independent analysis combined with the database search, de novo search, and then spectral library dependent improved the amount of sequences, the identification of the immunopeptidome. And actually, I just have to say that I'm very confident that a combination of data spectral library independent together with FASTA against FASTA, so spectral library independent analysis of DIA would help to have a higher accuracy and a better improvement of the immunopeptidomes. We have here shown that we are able to fit the spectral library we generated from our own proteome and peptidome from the control mice and OB mice. And the data proved that PICS was a reliable method to assign both the proteome and the immunopeptidome in this type of dendritic cell. So we are one of the few labs which actually by this time we made a correlation between the proteome and the immunopeptidome presented by MSC class 2 in the dendritic cells from this type of model mice which mimics metabolic stress. So at the level of the immunopeptidome, I'm not going to present here the detail. You can go ahead and read the whole paper, but we were quantified using both mainly the DIA at the level of MS1, and we had actually a very good readout, expected enzymes from different metabolic pathways and markers of atherosclerosis were very well represented and enhanced, presented as peptide on the IAB-MAC class 2 in the OB as compared with control mice. And we also did a very good post-translational modification analysis on the MSC class 2 itself, IAB. So we were able to identify all the redox-mediated PTM on MSC class 2. So by this time we knew that both the proteome, the late endosomal compartment, MSC class 2, IAB from the OB mice as compared with control were heavily modified with different type of redox PTM. And that's we were arguing, can we actually find an assay which would actually ex vivo prove that selective antigen can be processed differentially by the late endosomal compartment from the OB or high-fat, high fructose diet mice as compared with control. So this is actually the assay which help us to respond to that selective question, if indeed the late endosomal compartment were losing the capacity, or they were becoming actually better processing some antigen, probably due to the unfolding state of the protein antigen due to the accumulation of PDM. So you can do this assay starting different mice model of different disease or the human blood. You can purify dendritic cells from the mice, or you can purify dendritic cells in the case of the human from PBMC. And you can purify the late endosome later on a purple gradient, incubate the late endosomes with a native antigen, filtrate the peptidome after 3, 6, or 12 hours. You have to titrate the time, and you have to titrate the amount of proteins and the amount of late endosomes. So you end up to have this pool of peptides with less than 10,000 Dalton molecular weight, which you can screen them later on, as you can see here. And further down the line, you can understand that this assay was originally developed to better understand the molecular basis for the having a differential display immunopeptidome under metabolic stress than in the steady state physiological state. So what I am claiming here is that the immunopeptidomes, even they are sequenced at high resolution, they may not give you the whole idea of why they are so different. Let's say type 2 diabetics and MAC class 2 immunopeptidome, MAC class 1 presenting cancer, different type of tumor antigen. So what we are proposing is that this assay, in which you have late endosomal compartment processing different antigens, could be a complementary tool to better understand why the immunopeptidome have such a different, diversity and different biological, pathological state than in the steady state conditions. So as you see here, the steps which will have to follow up to end up in the end with the hotspot analysis, like seeing the sequence coverage for each antigen. What I want to say is that you can use native antigens and native state, or you can do chemical modification, like you can expose your protein antigen to methyl glyoxide, which it is used to mimic the oxidized sugar. So you can have a readout of different peptidomes derived under different condition and under different folding state for your antigen. Next, after you do the hotspot analysis, you can use the NextSpan MAC class 2 algorithm to predict the affinity. I can just show you here 2 epitopes. You can predict the affinity, and then you can follow up downstream with the in vitro analysis of MAC class 2 binding. You can use ELISA. You can use fluorescence polarization, and you can also use in vivo validation of understanding the immunological significance. Altogether, this type of analysis would help you to discover new epitopes, or epitopes derived from different antigens and the different chemical type of modification conditions from native state, as I said, to modified state. So why it is important? It is because you would generate a new pool of epitopes, which will be actually potential MAC class 2 binder. They could be further on tested for their immunological activity, as I have already specified. So this type of analysis was implemented using typical protocol in the peaks, which you see here on the left side, and in red, as you can see here on one type of antigen, model antigen, are, for example, epitopes we discovered not to be processed at all by the late endosomes in isolated from the OB mice. And the typical processing of the data is described in the paper, so you can follow up and develop your own assay based on your interest. I just want to show that in addition to following up the PICS protocol, we can also easily monitor actually the epitopes. You can see them in red here on the 2D map display on the PIC readout. You can see the specific epitopes derived from the antigen as compared to the other peptides, which are of course derived from the late endosomes, because you have also late endosome proteins which will be digested at 37 degrees. So you can export then the area corresponding to the typical antigen. We use here a model antigens such as valbumin, and you can see that the processing of valbumin peptide, which is shown up here on the screen, is different in the control mice late endosomal compartment than in the OB late endosomes. So altogether, the label-free quantitative analysis also in PICS enable us to quantify the peptide epitopes derived from the antigen processing. And I would say that this was a very good experience, and I will not gonna go ahead and present the rest because the time constraint, but those who have some questions related to this assay after you read the paper, you can go ahead and ask me, just send me an email. Altogether, this was not the first time when we use this assay. So you can see here in this slide, typical peptide epitopes, which we identify in another immunopeptidome screening and peptidome of the lymph. The paper was published in 2016. By that time, we were using PICS 7 and 8. So we map a high resolution, another immunopeptidome eluted from DR1 on transgenic mice, and we implemented as a pilot experiment that late endosomal processing to understand if a selected peptide epitope displayed here on the screen, it is processed by late endosomes or is processed by enzyme present in the sera. Like complement I was identified to be potential enzyme which is cleaving this peptide epitope from complement 3. However, the assay which implements the endosomal compartment and pure cathepsin, recombinant cathepsin, were shown not to have this peptide processed by the late endosome or by the cathepsin. You can see here nicely in this Venn diagram that the late endosomal assay was validated for the main recombinant cathepsins, and by that time we knew that we were gonna further on develop down the line for understanding the antigen processing. So I want to say that as a conclusion, the main findings of this research, if you read both papers, shows that indeed the late endosomal compartment are having a different capability to proteolytically process different antigen under different physiological state, and you can monitor the sequence coverage and quantity of different epitopes. You can discover new epitopes. You can also have different animal models knockout for different enzymes in the late endosomal compartment, and you can use this assay to identify the main epitopes derived from different enzymatic activity. There are some limitations, and we discussed this, and the titration is required to be sure that you don't over-digest the protein antigens. So altogether, thank you very much. And again, thanks to Columbia for providing the raw files, and to Dr. Stern for contribution to the Immunopeptidome, and thanks to the bioinformatics solutions. Any question, please?
Great, thank you, Christina. So there is a question here. It says, what is the minimum amount of cells required for an immunopeptidomic study, and when there is a limitation of starting material?
Yes, thank you for the question. It's a very good question. I would say that in our case, we were able to have at least 20 millions, but if we can get 50 millions at the minimum, and of course, 100 millions will be better. I mean, it depends on the, type of cells, but yeah, for the dendritic cells in the mice, I would say at least 50 millions stacked up.
Great, and then are there any other questions for Christina?
Can I ask one, Catherine?
Yes, absolutely.
Okay. Actually, I had two questions, but we'll see how we go. So I guess as we're going to hear probably later today, there are a lot of non-canonical sources of antigen as well. So I guess it becomes really important to validate your peptide identifications using synthetic peptides if possible. Are you able to generate synthetic versions of these glycated peptides to sort of validate that you've actually got the correct identification?
Oh, so meaning you want to say like synthesize the epitopes and then test them? We did that. Yes, actually, I'm sorry, I thought that I'm quite over time. So we really get here the epitopes, which were derived from ovalbumin, and we predicted them to be having selective affinity tests for the MAC class 2. What you do actually, we didn't spike. I understand. We didn't spike. You can spike with heavy label peptides and quantify them. We did this with other epitope, and our paper shows the epitopes from ApoB100, the Immunopeptidome. The ApoB100 was quantified using parallel reaction monitoring. So we heavy label the selective epitope. I can just show you here this one. So we have this epitope YSGS VAN, and we quantify using the PRM. We spike it over all the samples from the OB and control mice, and yes, we quantify, we prove that is increasing the OB than in the control. With respect to the late endosomes, like yes, you could spike the late endosomes too and do the same as I did for the total immunopeptidome. And the only way we could show actually in the paper that these epitopes, which are based on the late endosomal processing better, shown in the processing by OB than in the control, was to do a typical immunization. So what we did here, we just show that if you get the ovalbumin and you do the immunization with 100 microgram ovalbumin, you can see here that the CD4 cells were actually presenting not statistically significant, but they were enhancing in the T cell response for ovalbumin peptide as compared with the control. We use another antigen, HEL, and the late endosomal processing for this type for HEL was exactly different than for the ovalbumin. So we actually show this in vivo. But I agree, I mean, we should actually spike the late endosomal processing reaction mixture.
Okay. And the other question I had was around your observation that you see glycation of the class 2 molecule itself, IAFB. Do you know whether that IAFB is actually expressed on the cell surface with those PTMs? And if so, what do you think that means for T cell recognition of those complexes?
I am, I also have some reason, I show this data is because indeed it's very hard to argue that you will have statistically significant MHC class 2 on all the dendritic cells showing up with all these ppm. You have to know that these ppm are quantified from the peptidome and from the total proteomic analysis. So it's a combination of what you saw in both. But we never saw on the cell surface, to have an antibody selectively for IAB with a ppm. If that it is your question, I don't think there is something yet. What we can do, you can show total advanced glycation product. There are antibodies, very hard to find them, and you can show the total amount of AGE, but you cannot show specifically for each of these ppm. This is coming only for mass spectrometry analysis. Is that your question? What's your question?
I guess I was wondering whether these modifications prevented maturation of the class 2 molecules. And whether you see a decrease in class 2 expression on the surface of these dendritic cells relative to, I guess, non-stressed mice or a different genetic background, or whether the modification could exist on the cell surface and therefore impact T cell recognition of cognate complexes.
Yes, it's a very good point, but based on what I see, I'm showing here from other studies and my hours, is that CD11C still was significantly, the dendritic cell were upregulated, and what I don't show here, but actually we have proved that MHC class 2 in some cases remain upregulated proportionally with the amount of CDC11C. So yeah, this is just the dendritic cells themselves. It doesn't show the MHC class 2 upregulation. That's a very good point for a cellular biology type of analysis. Still, we have FACS analysis, and MHC class 2 was upregulated for some of them. But yeah, I don't think that we yet have a full analysis on MHC class 2 upregulation, how the PTM affect the MHC class 2 expression. Even in this cartoon, as you can see it here, you can see, this is from a review. It is suggested that you may have, it's always increased under inflammatory condition. We know that, but we don't know the total. The way the immunopeptidome we are doing is actually we quantify via ELISA to start up with the same amount of MHC class 2 and for the elution. So in some cases, if I go back in time and discuss with the lab of Dr. Stern who did the elution, yes, I think that there were some preps in which MHC class 2 were lower amount than in the control. But I have to review those data with them.
Thank you.
Great. Okay. Thank you, Cristina, again for your talk. It was really great to hear from you. And next up, we have Dr. Andy Yu Chi. Andy, I think you should be able to just share your screen now while you get set up. He is the lead proteomic scientist for the iPSC Neurodegenerative Diseases Initiative at the National Institutes of Health, National Institute of Aging. Dr. Chi received his Master's of Science in Pharmaceutical Sciences at Tianjin Medical University in 2012. He then went on to receive his PhD in Pharmaceutical Sciences at Wayne State University in 2017. And in his current role, Dr. Chi works on combining multi-omic approaches like proteomics, bulk single cell transcriptomics, and to understand the molecular alteration in iPSC-derived neurons harboring more than 120 Alzheimer's disease-related variants. And actually, very recently, Dr. Chi published 2 papers in MCP and cancer, where it was demonstrated that de novo sequencing and the pan-PTM search could yield long non-coding RNA-derived immunopeptides. And to validate this, T2 cell binding assays and synthetic peptides were used. So Andy, whenever you're ready.
Thank you so much, Catherine, for your kind introduction. It's really my honor to present our work here, and thank you, Catherine and Paul, to give me this opportunity. So, as I Catherine, you did a really good introduction about my current job, but I'm going to present some of my previous work. So, great. So here we go. I'm trying to advance my slides. All right, so some disclosure. So what we show here is we have I have no financial conflict of interest with any company, including PICS. And this is pure scientific talk. The views expressed herein do not imply perspective and the policy of the U.S. National Institute of Health. So I would like to start off with our a review has been written about 2 years ago. So we are from this review, we basically lay out the perspective of application of mass spectrometer based proteomics to identify the new antigen. So generally, from the generally people purify the HLA peptide from patient tumor or primary cells, and this is just a pure traditional pull-down analysis. And the trick is, as mentioned by Cristina, Dr. Cristina, is start with relatively larger amount. We start about like 100 million cells, for our tumor tissue, we start about 20 to 30 milligram frozen tissue. So the purified peptide is subject to the LC-MS analysis. And then the key part for the I was asked multiple times with what is the proteomic genomics. From my perspective, it's kind of a catchy word because basically proteomic genomics is just combine these 2 techniques. We use the genomics analysis to unveil the mutations or variants from the patient, and then incorporate those mutations to the reference database. And then we search the MS raw file against that customized, or we call sample specific or patient specific library. And then the following is basically just a traditional proteomics workflow. But by nature, increasing the database, actually, you're going to increase the false positive rate, as Paul also mentioned. So from my perspective, any peptide has to be validated by the targeted proteomics and follow up with immunogenicity validation, because that is really the key to call one particular peptide a new antigen, because that word should be used very careful. And then typically the workflow here is actually our current work going in progress right now. So the new antigen should house with the patient itself antigen presenting cells, and the co-culture with the tumor infiltrating cells isolate from the PBMCs. So if you're lucky, you can find a specific TCR against that new epitope. Then you separate out these T cells or TCRs and in vivo amplify the T cells and infuse the cells back to the patient. So that's we call adoptive T cell therapy. So this is the whole story. But if you want to close the loop, actually the majority work, actually, to be honest, it will be spend a lot of time actually the immunogenicity validation. But our work actually currently focus on the front end. So as Dr. Cristina just introduced of MAC molecule, so our lab actually currently focus on the MAC class 1, so which is a little bit different from class 2, because class 2 actually target on a CD4 T cell, which also have a longer epitopes. So for MHC class 1 is a shorter epitope, typically from 8 more to 14 more or 13 more. And the cells has to be, the epitope has to be processed endogenously and presented by the MHC molecule, class 1 molecule, and recognized by the CD8 T cell. Hopefully that some of the T cell receptor can recognize the epitope and eventually eliminate the tumor cell. So currently there are 2 class of the tumor antigens. So the first class will be tumor-associated antigen, which is existing in tumor and germline cells. So from my perspective, actually, this category has been well overlooked. So our experience in the germ cells, actually, the HLA expression is relatively low. Many of those tumor-associated antigen, where we can call it cancer testing antigen, are very, very actually active. But historically, this antigen all like being identified in melanoma, you name it, like GP100 or Prime E and NY-ESO-1. So, but they are not really working great from the clinical perspective. But I think this category should be really emphasized in the future. And obviously, the second category is more interesting in majority of the community. So tumor-specific antigen, which is like as is the name of it, is just specific to tumor, not in the normal tissue. And normally those antigen contains a somatic mutation, which is really the key of this entire processing, because somatic mutation actually unique to tumor, right? So our lab has long-term interest in the patient with mutated EGFR lung cancer, but unfortunately, those patients shows really poor response to the anti-PD-1, anti-PD-L1 therapy. And when we're looking at those, why the patients do not respond well to the checkpoint inhibitor therapy, because one of the review paper mentioned the PD-L1 actually is low expression compared to in the EGFR mutant cancer versus the EGFR wild type. And more importantly, the tumor mutation burden actually is also significantly lower in the EGFR mutant lung cancer compared to the wild type. Therefore, our lab designed using this model or using this opportunity. Actually, we're trying to build a comprehensive immunopeptidome profiling specific on class 1. So as you can tell from the cartoon, actually the upper arm is proteome part I just actually described. So basically purified HLA peptide and sent to the mass spec sequencing. Nothing fancy, but we just want to point out, because many people mentioned the DIA. So in this particular study, we use the DDA search. And then the lower arm, we use the genomic DNA and RNA doing the whole exon sequencing, which we use for the HLA typing, which is so important to actually predict the peptide to binding to which allele, because we all know HLA or MHC, they're highly polymorphism, so many variety of the HLA. So, and the different HLA peptides could bind to many different epitopes. So, and then we use the DNA sequencing, also called the single nuclear variants or SNPs, and also the small indel and the deletions. And from RNA-seq data we use for the called the gene fusion. We use an informatic tool developed by David Fanil in the NYU. So this is a really useful piece of informatic tool that you can basically do in silico translation from your RNA or DNA sequencing file directly to a protein FASTA file. And then for this particular pipeline, actually, I'm gonna emphasize 4 different categories. One is for the mutant peptide derived from the somatic mutation in tumor. Second category will be the cancer testing antigen. And also I want to touch upon a little bit on the PTM peptides, also link RNA derived peptide. So as like sort of, we want to QC our data, whether we are finding the actually class 1 peptide. So that's the first thing we did. So we were able to identify more than 20,000 peptides, and as expected, 9-mer is the most dominant peptide in the entire immunopeptidome, which we have higher confident. Also, we did motif analysis. They are all aligned with the known alleles. And then, so we were able to find 12 mutant variant peptides from 2 EGFR mutant lung carcinoma cell lines. So they are from 9 more to 11 more, and each of them has at least one respective allele being predicted to be a strong binder by NetMSC. This is a computational algorithm commonly used in this field. And also, of course, we use synthetic peptide to validate actually 11 out of the 12 epitopes. This is very important to mention here actually from the mutant peptide, we're able to find this 5 of them actually being reported somatic mutation, and also you can see some of the epitope not only reported in lung cancer, also in blood cancer, pancreatic cancer, and colon cancer. So, which means any of the epitope you have, if the patient carry the right allele with the correct mutation, actually we can develop a T cell against that. So this is really the dream for us. We're ultimately go for this entire study to develop a TCR to benefit the patient, benefit the entire community. So since I was asked for doing this talk for introduce a peak, so I really want to emphasize I was really happy with the peak search. So I will show some of the result here with especially with the de novo search. That was one of the main reasons we picked peaks as our main search engine. So as you can see here, the database search result, we use NetMSC prediction algorithm. Any of those, if they are predicted to be a binder or percentage rank less than 2%, will be labeled as a binder. So we were able to find 60 to 70% of the peptide in the database search, and compared to the de novo search, we have to admit that without a targeted decoy database, there's no FDR control. So we are able to about find like 30 to 40% of the peptide actually predict to be a binder. So it's reasonably high compared to other searching engine. So, and also actually Paul just showed this very similar. We did the same thing. So from the motif analysis with the database search and the de novo search, we see these two actually the motif was for one of the PC9 cell line is really great align, especially in the number 9 anchor position. So they align really well. And okay, so this is one of the basically, we really want to try the hydrophobicity and the retention time correlation. This showed first figure actually from one of the comments of our Tony Purcell, Dr. Tony Purcell's paper in Science, very famous paper addressing the spliced peptide with cis and trans. So the comment they actually showing the spliced peptide was not really correlate with the hydrophobicity index, but there's a lot of things going on with this comment. So, and our Dr. Tony Purcell addressed really well, in my opinion. But we still did that for the sake of the reviewer purpose, whether they're gonna ask us what about the retention time and hydrophobicity, which is the key for the chemical property of each immunopeptide. So the database search, we find about the correlation about 0.87, and for de novo only, we find 0.66 correlation. So definitely, I think it's really reasonable, as we saw before. It's definitely a key to actually to see that de novo only search actually has result reasonably high confident, even though you don't have any FDR control at this moment. So then next we asked, this is really interesting. Actually, one of my students asked me like, hey, if you use the standard human proteome reference, if you think the de novo is so good, whether you use the standard human proteome search, and then whether you can retrieve the peptide that you use a patient or sample specific library. Then we did that, and we actually retrieved the peptide from the de novo only search using standard human database without adding any mutation. And the middle panel actually showing the database search peptide, we have the mutation actually incorporated into the library. And the last panel is the synthetic peptide. You can see the spectra and the retention time, they align really well. So we were able to find 4 out of 7 in this particular case being identified in the de novo. And also there is another cell line, the PC9, we found 2 out of 5. So we are very happy with the result actually. So using a de novo, we can retrieve more than half the peptide, the mutant peptide in this particular search. So when we did the correlation with the HI, the hydrophobicity index and retention time, we got amazing R-square 0.99. So it's a really good indication of the de novo search has done an amazing job here. So I want to switch a little gear on the cancer testing antigen. So be honest, that's not much we can highlight here because this basically is a traditional database search. The only thing I want to highlight, we spent a lot of time with a collab with the Javikan in the NCI genetic branch that we built a high confidence library built on 3 known database, human database, and then it's about 255 of the tumor testing antigen or overexpressed antigen has been including in this. So when we identify the wild type peptide against this library, we were able to find 40 of the peptides belongs to, cancer testing antigen assigned to 14 protein assigned to 14 proteins. And the 5 of those are novel, hasn't been reported in the IEDD. And I want to highlight that actually the one particular peptide was identified in the lung cancer cell line, whereas hasn't been reported and showing really great efficacy actually in the lung cancer. So we also use the synthetic peptide for validate some of the peptides. Just too many, we didn't validate everyone. So for PTM search, and Dr. Cristina also mentioned this, for PTM search peptide, we call this pan PTM. Basically, you select that, you click that button, the box in the peaks, so you can show you like we do all the PTM without doing the individual search. So we're amazingly to find more than thousands of PTM peptides, but this is just a search. We don't have too much thing to highlight here, but I do want to highlight one thing here. So because the speed of searching the pan-PTM using peaks is just day and night different compared to using other searching engine. If you ever put like 4 variable modifications in a mass quant with no enzyme search, and maybe like 40 or 20 raw files, you probably just kill your computer. I mean, it's probably take weeks to finish. And the PIX is very fast to finish the whole thing. And I think the main thing is for PIX using a sequence tag-based search. That was, I think, the very beginning was invented by Mattisman in 1994. And then on the other hand, the MassQuant, MassGauss, Sequest, or PD, they use a library-based search, like John Yes, I think the first one invented in 1995. So it's a really fast search. I mean, it's just unparalleled with any other searching engine to doing this 9 enzyme and the PTM search. So one thing actually, Dr. Tony just mentioned, actually, we also did the PTM search. And one example here, what we found here, there's one modified peptide being identified in 2 different types of samples. It's ANX, A2, so it's not a very critical protein anyway, but this peptide was able to find in 2 different forms. One is deamidation, the other is methylation. But when you have the modification, they only identify either in one sample, the other in the methylation in RA07 is a tumor tissue. So which means I will assume if the modification happened both in both samples, because both are lung cancer cells, they probably share very similar mechanism. And then if you present completely differently in the PTM version, so which is good indication that this could be affected by the binding affinity with different alleles. So for link RNA, for non-coding RNA, so here we actually a little bit different than the traditional way doing this. Probably Dr. Tony can comment more later. So we use the Linkipedia database. Basically, we know the HG38 or standard, reference does have a lot of link RNA being annotated a lot, but we're thinking is like, probably we want to have a better one. So then we find the Linkipedia is really reliable source with all the link RNA being annotated in this particular database. So we use our RNA-seq FASTQ-C files to actually align not to the HG38, but for this link RNA reference. And also we leverage the de novo search actually because we know the de novo only peptides, which means they're not aligned to annotating any of known database, including the mutations we identify from patients. So this must be some from intronic region or non-coding region, right? So then we were able to find 71 of them when we do the heat map, then showing actually a lot of those not being expressed, but 53 of them being expressed at the RNA level. But this is the full length of link RNA, not for that particular peptide. So when we're actually showing the peptide, we show 44 peptides. When we look at every single one, they are actually, all the peptides are unique to each sample in the patient. There are no overlapping at all. So we're surprised because we saw that actually there are many of the peptides being, at least full-length link RNA being annotated in the RNA-seq, but it's not really show up in the peptidome because our explanation was the HLA actually allele different HLA restriction actually is probably play the key here because of the different allele, they may present different antigen. So, and then the common antigen actually, a common oncogene actually is probably the most attractive way to doing the tumor, to be as a new antigen. And we were able to find many of them as really known oncoantigen, KRAS, EGFR, P53, those are wild type antigen, but one thing we want to point out, very similar, we saw only one actually antigen share between two samples. They're very, very rare. You can have one antigen actually share with different patients. So that's leading us to our actually current study. We really want to find that common oncogene derived new antigen. So this is a shared somatic mutation that can be shared with multiple patients. So we're not be able to develop a T-cell therapy for every single patient. I mean, it could happen, but I mean, it's going to take more effort. So if we have some off-the-shelf TCR targeted any of the common mutations, so which is really the dream for this entire field. So what we did approach with to use the monolithic. There's only one HLA allele expressed in the K562 antigen presenting cells with the transgene of the tandem milligen. Tandem milligen actually contains different common mutations, about like 20 more with the sliding window. So we're going to screen that the new antigen by shotgun proteomics, and then the one we find if it's positive, we're going to buy the heavy synthetic peptide and spike into the IP sample and do a 10-minute gradient for a ISRM assay, and we monitor 5 to 6 transition of each peptide. So currently we find the KRAS G12V, which is also very common mutation among many solid tumor. So this is the spectrum of this particular peptide, the mutant peptide actually being shown in peaks from the DDA data. So very strong signal. So we were able to make the heavy label peptide and make you can see a really great co-elution, and in 2.7 minutes, you can see this peptide is really great and it's high throughput. We can screen many patients in this way. This really have a lot of translational medicine meaning. So, but data has not been published yet. So currently we're doing the TCR screening right now to really want to find I think we have 4 TCR already, but I think one really have great efficacy right now, but it's too early to tell this is going to be a succeed. So at last minute, I want to promote our 2 presentation in ASMS in the coming week. So one is for the understanding of the molecular mechanism in the osmotic resistant EGFR lung cancer. So this is basically a follow-up study of our previous publication. So this is, we make the select label peptide because in my opinion, actually, label-free probably not going to be the best way to quantify. We like really quantify the immunopeptide because there's so many steps in the sample prep, and then many of those could go wrong with human error. So to eliminate the possibility systematic error, I think it must have to be metabolic labeling the cells and pull the cells, and then every cell line goes through the same process to eliminate the potential issue. So we did from the same cells, we did HLA peptidome, we did whole cell proteome. Also, we also put down the HLA complex, and then we also profile the interactome. So our finding mainly find the immunopeptidome actually is very significantly decreased, and then we find many of the HLA complex and the proteasome complex has been reduced reduced the regulation and downregulated in the alpha-protein resistant cells. But if you want to learn more, come to our poster on Tuesday. So another one, I will give a oral presentation with my current research. Really mainly my main current research doing cryptic peptide, a biomarker in the aging disease or neurodegenerative disease. So it was a frontotemporal temporal dementia. So those people, when they have to actually reach to the disease, have a dementia, they start to, lose their speech, or they start forget their name. So it's very late. So we want to find a early onset biomarker for this really vital disease. This could happen not only in aging people, it could be happen like young people, like 20s. So the key here, so one of the key pathology genes called TTP43, when we saw from the patient brain, this TTP43 knockdown or TTP43 loss actually is very early in the patient. So we make a iPSC stem cell derived neuron and knock down TTP43. We saw there's a cryptic exon. They basically is intronic. In the intronic region, they basically have expression between, for example, exon 1 and exon 2. They have something here in the cryptic exon. So the goal is to detect the biomarker encoded by the cryptic exon. So we have have great success with using peaks, very similar pipeline, and we were able to find 50 peptides for now, coded by the cryptic exon, and we were able to find 22 of the peptide has been validated by the highway label peptide. So if you want to learn more, come to my talk on Thursdays, November 1st. So the take-home message, hopefully I've been convinced you that the proteomic genomics combined with peaks provide really a comprehensive platform in the new antigen discovery, and the peaks de novo search is really a reliable tool for any of the downstream analysis, and the pan-PTM search is really quick. So, and then also it's good for large scale profiling because other search engines just cannot do that. We also provide a large database with 250 tumor-associated antigen, which could be off-the-shelf database you can use to search your current immunopeptidome data and try to find some cancer testing antigen. And the last one is more important, actually, our ongoing project with the surgeon branch with Jim Young and Steven Rosenberg to find the common mutant oncogene derived neuron. Derive the epitope, whether those are probably the final solution for the many patients suffering with the multiple different solid cancers. So with that, I want to thank my colleagues at NCI. He was Udayan Bukhar is my postdoc advisor. And from Javid Khan is our main collaborator on the bioinformatics part. And from Surgeon Branch, actually, they did a lot of on the T cell work, David Fennew. And also my current lab has the many colleagues need to be a really great support my cohort, and this work was actually the new project. The biomarker was collab with people from Mayo Clinic and Emory. So our funding is from the NIH intramural program. And the last thing I show the figure here is the building 10 of NIH campus. NIH has 50 buildings. The myth is, before people ask me why NIH doing research, but actually NIH not only the agents spending the distribute the money from the Congress, actually NIH doing really good research by themselves. So we have intramural program, and it's about like 1/10 of the total funding go to NIH itself. So we have really large campus with about 20,000 PI and the trainees. So yeah, with that, I would like to thank everyone's attention, and I'm happy to take any questions.
Great, thank you, Andy. Is there any questions for him?
Start off with one, Catherine.
Okay.
Thanks, Andy. Interesting talk. I think we're in agreement that the Novo tool works really well. I think we'll see my talk a little later today. My question, and maybe it's even more of a comment. So looking at the PTMs that you were profiling, I guess the one that was really missing were phosphopeptides. So I'm guessing that you probably do a single LC-MS run for your antigen discovery work.
Yeah, actually, yeah, the majority of the work was done by a single shot. Yes.
Yeah, because the comment I would make is that so we tend to do fairly extensive fractionation of the material, and there you do start to see a lot more of the different PTMs, and particularly the phosphopeptides, and that's without phosphopeptide enrichment. So, you know, I guess particularly in the context of cancer, obviously those types of antigens are of great interest as well.
Yep, for sure. Yep.
Okay, there's also another question here, Andy. It says, how can we validate PTM immunopeptides identified with PEAK's pan-PTM search? So example, like if all the peptides identified might not be immunologically relevant.
Yeah, that's a really good question. Actually, if you want to validate the immunogenicity, actually, basically you have to make the peptide and the co-culture with the T cell. So I think that's the only way you can validate that. So with current pipeline we develop is basically just profiling. We can prove the direct evidence that the peptide exists, but we're not be able to say this is actually immunogenicity cells, I mean peptide epitope. So it's a lot of work need to be done. So
I have a related question. Great talk. Thank you, Andy. You know, the question is like, if you really want to validate, like Dr. Tony Purcell said, if you want to validate this PTM on the peptides with PTM and their immunogenicity, I know that there are companies which are can synthesize actually phosphopeptides. If you really are sure about, you know, the site, you know, how well it is identified. So phosphopeptides are easy to synthesize, some acetylation and that, but like as I have shown, those which have a selective redox PTM, you may never get easy synthesized such as peptides. So like those complicated adducts of arginine or lysine and oxidized methyl glyoxide are not easy. So I don't know, did you try to validate in the T cell assay any of your phosphopeptides or deamidated peptides or anything?
no, yeah, that's a very good question. Actually, one of our reviewer asked this question. So as you mentioned, actually, one is the cost. So it's very expensive to make any of the PTM peptide, and also we are worried about the purity actually. So if they make any peptide with a large amount contaminated with the unmodified peptide, it will be not really helpful. So and also with the timeline, so it doesn't fit our timeline either. So that's why we didn't do any of this.
Great. And is there any other questions today for Andy? Okay, well, if not, thank you, Andy, again for that great presentation. And then, so next we have Dan Maloney. And Dan, if you'd like to go ahead and share your screen. Yep, perfect. Those who are familiar with peaks will most likely know Dan as well. Dan has been at BSI for over 12 years as one of our application scientists. And it's unfortunate he's not going to be attending ASMS this year because I think that's how many people end up finding our booth. You end up seeing his head over all the other ones. And I mean, in addition to providing support at BSI, he has also has an research interest in the regulation of protein activity. He's currently collaborating on a research project at the University of Guelph to discover the regulatory pathways involved in starch synthesis. Today, though, as you can see from the slide, Dan's going to be giving us an overview of Peaks Online, which is a high throughput solution for data analysis. It's been growing a lot in popularity, especially with the increasing file sizes of mass spec data today. So Dan, over to you.
All right, thanks, Catherine. Thanks for the introduction. Am I sharing? I'm actually sharing the screen. You just see the slides right now. Is that?
Yep, just this.
Okay. All right. Thank you. Okay. Yeah. Hi, everyone. So today I'm going to be going through an overview of Peaks Online. So I know that quite a few of you in the talk might be completely new to Peaks. So I'm going to be giving a bit of an overview of Peaks as a whole for the first 5 minutes, just to give you an idea of what Peaks is all about. All right. So first, I'm going to be talking about Peaks, the algorithms behind Peaks, and then I'm going to go into an overview of what Peaks Online actually is and what are its advantages for high throughput analysis of protein identification quantification. And yeah, then I'm going to go through an example application of label-free quantification with Peaks Online. All right, so what is Peaks? So Peaks was originally designed as a de novo sequencing tool. And from there, we've started to add functions on top of that ability. So such as PeaksDB, which is a de novo assisted identification tool, as well as Peaks PTM, which Andy so kindly described pretty well, and Spider, which is good for finding point mutations. In later versions of Peaks, we've also added the ability to identify peptides from reference spectra. So basically that means a library search, and that was designed to deal with complex situations such as DIA data sets. Yep, and also we have our quantification tools, so label-free quantification or SILAC quantification, which are based primarily on peptide feature detection. And I'll go through that a little bit, as well as quantification of isobaric labels such as TMT or iTrack. All right, so de novo sequencing. So to give a very brief description of what de novo sequencing is, the concept is to predict what the most likely peptide sequences that matches up to a spectrum from all possible peptides that match up to the precursor mass. So a pretty difficult task. But what Peaks is able to do this pretty quickly and accurately by looking back from the precursor mass and forward from 0 in both directions simultaneously to try to predict which peaks in the spectrum actually match up to your actual peptide. So the end result of this is that you get a spectrum match such as what we see here, where red peaks are matched up to X, Y, or Z ions, and blue peaks are matched up to A, B, or C ions. And based on how many peaks actually match up to your actual predicted peptide, it'll actually do this thousands of times with all the different possible matches that can match up to your precursor mass. It will try to predict which peptide is the best match to the spectrum based on machine learning using known data sets. And then we give a result which is the local confidence score, the average local confidence score for all the amino acids in the peptide, to signify that we are confident or not confident in the peptide sequence. So using that ability to actually predict the best peptide match for a spectrum, we can then do more things such as PeaksDB. So the concept of PeaksDB is that it's a de novo assisted database search. So we look not only at the peptide from a de novo perspective, but also by searching with a protein sequence database and try to find what is the best possible peptide match to the spectrum using two different techniques. And the simple logic behind that is if you have two different techniques that are trying to come to that same answer and they come to the same answer, that answer is much more likely to be true. So if the de novo is similar to the database search result, we'll increase the confidence in the de novo sequencing in the database search result. If they're very dissimilar, that answer is much less likely to be true, so we'll decrease the confidence in that. And if you look at that in terms of a traditional false discovery rate curve, here in blue are target peptide hits, and in pink are decoy peptide hits from a randomly generated decoy database. We are able to separate these two distributions much better by including de novo sequencing in our scoring algorithm than if we weren't using de novo sequencing. And then beyond that, we also have our Peaks Vtm tool. So what this does is it takes unmatched peaks spectra that are unmatched by PeaksDB, and also those spectra must also have a confident match by de novo sequencing. By doing that, we're able to reduce the search space of spectra to only ones that are unable to be matched to a database easily. And then from there, we're able to search for all of the naturally occurring modifications in the Unimod database, and then return not only your unmodified peptides, but also any unexpected modified peptides as well. And then we also have our spider search tool. So the spider is actually closer to a blast search than it is to a traditional database search. And the concept of this is that we search through your target database and with your de novo sequence tag until we find a sequence in the database that has a similar sequence to the confident part of your de novo result. So this is represented here by red letters, meaning that we're above 90% confident in this part of the de novo sequence. And you can see that in the database, we have a pretty close match to this sequence, exception of this alanine in the de novo result. So what Spider will do is it'll generate a Spider candidate, which is a combination of the database and the de novo sequencing result, and then do a spectrum alignment of that to see if that's a good match to the spectrum. And if that's a good match to the spectrum, it will then return that as an additional result in your identification results. Yeah, in more recent versions of Peaks, we've included a spectral library search. So what this does is it takes a good fractionated, generally you want to use a fractionated sample as a reference proteome, and you generate a list of spectra that are confidently matched to your protein database. And then it will generate a library spectrum from that, meaning that we take the matched ions as well as some extra ions as well in some cases, and generate a text file which contains the ions that are matched, their M/Z intensity, as well as some extra information, including their retention time, their indexed retention time, as well as ion mobility information if it's from an instrument that's able to support ion mobility. So then what we do is you generally take a DIA data set and search that against your spectral library. And generally speaking, DIA spectra can be quite complex, which means that just trying to match up the fragment ions to your library spectrum can be quite difficult and not going to give you an accurate result in some cases. So you need to rely on some extra information. So for example, you could lean on the concept of retention time prediction, where we take the retention time of our spectra in the library and compare that to the actual retention time that we observe in our DIA data set and create a regression showing whether or not the expected retention time is matching up with the actual observed retention time, and using that as part of the scoring. As well as we look at the distribution of the fragment ions across the retention time range and look at the precursor peptide profile. And the result of that is you get a match between your spectrum, your actual query spectrum, and your library spectrum. Yeah, and then we also have quantification based on the LC-MS. So you often hear us mention this concept called peptide feature identification. So generally from mass spec, if you look at it in a 3D view, where you have the M over Z on the x-axis, the retention time on the y-axis, and in a 3D plane, we have the intensity of the signal coming out of the mass spec. A peptide will usually have a certain distribution in terms of its carbon isotopes, the carbon 12, 13, and so on, and a certain shape across the retention time range. So now Peaks is able to pick up on this distribution and then draw what's represented here, the shape around the peptide feature, and we calculate the area under the curve of those isotopes, sum them together, and give you the intensity of the peptide feature. You can do this across different samples and match them up. And then by doing that, you're able to get the ratio of that peptide across different samples in order to do a label-free quantification or SILAC quantification. In that case, it'll be within a single result. Yeah, and those are the main points that are included in Peaks Studio and in Peaks Online. So today I'm going to highlight Peaks Online for you. So what actually is Peaks Online? So Peaks Online is a server-based distributed system for high-performance computing. It's designed to get the most out of Peaks with some really good hardware. So it's designed as a simultaneous multi-user solution, meaning that several different users can send jobs to the Peaks Online setup, and it will be able to run those different jobs simultaneously. It's highly scalable, meaning that you can add servers to it over time in order to increase the power of your Peaks Online cluster in order to get the most proteins and peptides back in the quickest amount of time possible. So I would like to point out, based on the name Peaks Online, you might think that it's an internet-based solution, but you do have the ability to set up a Peaks Online server on your local area network that will be closed off to the outside world, or you could set it up in an optional cloud-based framework. And so the results that you then end up getting from Peaks, which I'll show in the actual live demo, it's all Peaks, but in a web browser interface. So you interact with your master node through a web browser interface. So you send jobs to that master node, then it will distribute the jobs to worker nodes, which will then work with the different resources that are available on the network in order to generate your identification and quantification results, and then give those results back to you through that same web interface. All right, so the advantages that Peaks Online is able to give to you is that it has unprecedented speed, and it's quite easy to use, and it's also automatable, which I'll go in a little bit of detail about as well. So first, about the speed. So here we have an example case where we have a huge amount of data, a 672 3-hour mass spec runs, 50 million MS1 spectra, 30 million MS2 spectra. So with our Peaks Online interface, we're able to complete this run in 230 hours with a 32 core setup. So still pretty quick compared to the amount of time that it actually took to generate this data set. But then as you increase the amount of processing power that you give to this setup, you're actually able to get it down to 17 hours if you're using a 512 core setup. So a pretty hefty hardware setup, but you're able to run through data through peaks at an alarming rate at that speed. Yeah, but also it's quite easy to use. So you're able to set up some predefined workflows. So you go in and you set up your parameters for your search, and then you save those parameters. And once you do that, you're able to set up a workflow. So you click and say, select a specific workflow, give the name of that workflow and click next. And then basically you're done, and you'll be able to get your results back once Peaks has generated them. And beyond that, it's quite easy to review your results and set up new parameters once you take a look at your results and decide, okay, I want to change the PTMs that I'm searching against, the error tolerances, that sort of thing. It's quite easy to just go in, click the Modify Parameters button, and it will, I'll demonstrate this in the live demo, and it will just recreate your analysis, and you just make the changes. And the important thing about that is Peaks will recognize the changes that you make and only redo the steps of the analysis that are necessary to get the new results back. So it'll keep track of the changes and keep what it can keep and redo what it has to redo. Yeah, and just to review the functions that are in Peaks Online now. So there's some different analyses that you're able to set up, and they're split into two different drop-down menus. So for your DDA data set, you were able to do your traditional label-free quantification workflow, which I'll talk about here, or you can set up with an identification workflow where you'll do database search, Peaks, BTM, and Spider, or some different other options here. If you're using a DIA setup, you have what Paul described in his talk, where you go through and do a spectral library search and then a database search followed by de novo sequencing or by quantification if that's your end goal. All right, so what about automation with Peaks Online? So one of the neat things that we're releasing in the next version of Peaks Online is the ability to set up an instrument daemon. So what does that actually mean? So basically you set up a connection to your actual instrument computer or any computer or any folder where the data from your instrument is actually going to, and it'll actually monitor the folder where you're actually kicking the results out from your instrument, and it will monitor that folder for any new data files. And you get those new results, it will actually be able to run searches as they come off the instrument. So basically the setup is like this, where you set up your delimiters, you give Peaks an example file name, and it will look for certain trends. So you could set it up to look for underscores, and you set up a project name based on, like, say, the instrument name and the length of your run. Also specify that what you use to actually name your samples. And you can even give information in the raw file name, such as your enzyme and activation mode. And then Peaks will keep track of what's being uploaded to that folder and set up new runs as they come in. But we also have a command line interface available for Peaks Online, where you can just set up your searches through a command line interface and automate it that way if you prefer. Yeah, so just a quick note about the consistency of the results from Peaks Online with Peaks Studio. So we've done some testing to make sure that the results from Peaks Online and Peaks Studio are always consistently similar. So looking at the same data set between Online and Peaks Studio, generally speaking, you're within 2% in terms of the number of proteins, peptides identified, and as well quite similar in terms of the de novo results. So I just wanted to mention that there. Right. Yeah, so with that, let's actually jump in and take a look at what the software actually is. So I'm going to be going through a live demo of a label-free quantification example. Just a pretty typical example that you often see these days, where you have a human yeast and E. coli lysates spiked in different amounts. So we have our 3 replicates of HYEA, and then we have 3 replicates of HYEB, where human is expected to be a 1-to-1 ratio. E. coli is expected to be a 4-to-1 ratio of A to B, and then the yeast is expected to be a 1-to-2 ratio between one samples A and B. Yeah, so this is Peaks Online here. So first thing, I want to show you, just give you a lay of the land of how to use Peaks Online. So when you come into Peaks Online, you'll have these options up here in the top right-hand corner. You can go to Settings, Databases, and then you can bring in protein sequence databases from this table here, or you can also bring in spectral libraries here as well. And if you want to set up a new search, you just go up to the top left-hand corner, go File, Create New Project, and then you can start to add your data. Yeah, so from here, if you had set up an instrument daemon, this is where it would show up here. You can see that you have some different options to load in data. You can just click this button to load in data directly from your local computer. You can click the remote button. And basically in this case, you would have set up previously some remote connections to folders on your network that actually have where you store your raw data. So I'm going to show here and show a little bit about how easy it is to actually set up a project with Peaks Online. So let's say you have many raw files that you want to load into your project, but you don't want to actually go through and set up all the samples and create new names for each of the samples. You can click this button down here at the bottom to create new samples by regex. And then you'll be able to specify what you want to name and how you want to group your samples based on the raw file name. So if I click this button to separate it by the underscore, I then notice how we always have A in this case to denote the 2 conditions, A and B. So you click that button here and click this button here to let it know that I want to include the replicate as well. Then you upload, and then it automatically went through and created 6 different samples based on the naming of the raw file with the appropriate name up here in the top left. And then you just let Peaks know the activation method, the instrument type, the enzyme, and then you just go through and click next. Yeah, and this is where you basically choose your own adventure. So you want to choose which type of quantification or identification based on the data set that you're using. In this case, you're going to do a label-free quantification and click next. Yeah, and this is where you set up all your identification and quantification parameters. But at this point, I actually want to switch and show the existing results. So I'm going to go back here to my projects and open the results. So let's say I've already ran this analysis. You can see we have a few different analyses over here on the top left, and I want to make some changes to the parameters. All you have to do is you click the analysis that you want to copy, click the modify parameters up in the top right-hand corner, and it will populate the parameters page with the parameters that you used in your previous analysis here. So of your error tolerances, your enzymes, target database, quantification here. And then, yeah, you could also set up your filters beforehand. Yeah, so notice here, you actually are able to set up your FDRs based on peptide, PSM, or by the score, and based on even at the protein level here, and your de novo sequencing filters. And you're able to do this all beforehand without actually ever having to finish result. Then set up your quantification grouping. So here I'm actually just going to show a bit of a new feature here. You can also set up your quantification groups by regex by clicking this button here, then set it up by the underscore. In this case, I want to group them based on A, so to see when there's differences in this element of the name, and then click save, and then automatically we just have two groups. So it picked up that there was just a difference in A and B in the raw file name here, so it was automatically able to create two groups out of 6 raw files. So you can imagine if you have hundreds of samples and you make sure that your raw file names are distinguished by that certain thing, it'll then be able to automatically create your groups based on your conditions without you having to go through lists of raw files and figure it out, which is of course tedious and also can result in human error. Yeah, and then you just click submit and it will go through and do your new analysis. So let's take a look at some results. All right, so here we have the results of Peaks Online. So the first thing that you notice is that the result view looks very similar to what you have with Peaks Studio. You have your summary page, protein page, peptides, the de novo sequencing results. And then here in your summary view, you have a breakdown of all the identifications that were made, number of peptides, sequences, protein groups per sample, and your false discovery rate curves, as well as some other statistical charts. So the next thing that we want to do is we want to go to the protein tab. And here you can see a breakdown of the different proteins that were identified. So you can see that this looks a little bit different. And here we're trying to show you a little bit of extra information. So you can see that in some cases you have a darker pink color for some parts of the coverage page. And if you look at the coverage legend up here at the top, this shows you how many spectra actually identified that part of the protein. So you can tell by the intensity of the pink color how many actual spectra identified that part of the protein. But then if you want a more traditional view, you can come here and take a look at click specified by each sample, and then you can see for each sample the breakdown in the coverage in terms of the peptides, where you see a blue bar showing which parts of the protein are actually identified by peptides. And then you can click on any of these blue bars and it'll bring up the best spectrum that identified that part of the protein. Yeah, and then I also wanted to highlight the de novo only tab. So this can be a very useful tab that's very unique to Peaks. What you get here is you get a list of all of the good quality de novo results from spectra that could not be matched by database search, meaning that these spectra could not be matched to any entries in the protein database, but they're still, as you can see, pretty high quality spectra. So you can use this as a starting point to try to find some additional identifications that you may have missed if you just focus on the identification results. Yeah, then let's look at the quantification results here. Go into the protein tab. And here you can see a list of all of the proteins that were quantified in our example. Remembering back to our case here, we have a mixture of human, yeast, and E. coli. And the idea here is that we have 3 distributions which are already quite visible here in the coverage view, where we expect to see the yeast proteins upregulated, not upregulated, but more abundant in our B condition, the E. coli proteins less abundant in our B condition, and the human protein should be remaining the same. Yeah, and you could see based on the heat map, the ratio just to actually highlight it in a way that'll be easier to see. This is a kind of cool thing that you can do, is you can click the protein filters button, put in, let's say, yeast, and it'll light up here nicely, showing all of the proteins that are coming from yeast here. Then you can see whether or not there's a consistent increase in abundance of those proteins based on the sample profile. Yeah, and that's just been a quick run through of Peaks Online. So all of these views are quite helpful. But then once you then get your results, the main thing is that you can get through to the export quite easily. So you just click on this export page and you decided that your results are acceptable. You just pick which result exports that you want. And then you can download a full result for all of your data all together, or you can download these by sample and then share with your collaborators. Yeah, and that's all that I wanted to share with you today. Thanks for taking the time. And if you have any questions, feel free to go ahead now.
Great, thank you, Dan. There are a couple of questions in the Q&A. Did you want to read them out, or did you want me to?
I think I can handle it.
Okay.
I actually don't know where they start. Can you?
Oh yeah, sure. Nope, I got it. For DIA analysis, can peaks identify fragment ions that suffered from integration interference due to co-fragmentation peptides that generate similar fragment ions? And if yes, does it annotate these cases and choose not to quantify with these, or just avoid presenting them?
Can I see that? So co-fragmenting. Yeah, so is this with DDA data or DIA data? Was that mentioned?
It says for DIA analysis.
For DIA analysis. I think Paul might be the best person to answer that question. Paul, are you still on the line?
I think he is. He's muted. Paul, are you still there? Okay, well, if anything, we can get.
If not, I can take a crack at it.
Okay.
So in peaks, our label-free quantification is ID directed. So basically, if you do have co-fragmented peptides, then peaks should be able to identify them separately, and also it's going to be able to detect the peptide feature. Hopefully they'll be far enough apart in terms of their M over Z that we're actually able to deconvolute the two peptide features separately and create two separate peptide features from that. So taking that into account, and hopefully it will also have two separate identifications based on the library search or the direct database search, it should then be able to get two separate peptide identifications and quantifications for those two different peptides. So I believe, yes, it should be able to do that, but of course that's going to be a difficult case.
Okay, great. And there's another question here. They say that they really like the tools of PTM profiling and multiple sequence alignment in Peak Studio. When could these tools be added into Peaks Online? And they would be really important for research about ancient proteins. I don't know if that's a question for Brenton and Lei, if they are on here.
And if not, you feel free to interrupt me if you guys do have an answer to that. But technically speaking, you can get all the information that's provided in PTM profiling out of the peaks result from the result tables, because basically the PTM profiling results is basically just a subset of the label-free quantification results. So in the label-free quantification exports, particularly in the PSM export, you're going to get, in the peptide or the peptide features, you're going to get your identified unmodified peptide, and you're going to get your identified modified peptides, and you're going to get the areas for all of them across all the different samples. So what Peaks PTM would do with this is it basically it automates the process of taking all that information together. But basically what you just want to do is look at the ratio between your unmodified peptide to your modified peptide and calculate the percentage of that. So it is technically possible from Peaks Online to get the same information that you have from PTM profiling.
Okay, and then last question I see here is, what is the difference between two similar settings or different settings of modifications between the de novo search and the DB search? And would the results be very different, or how different, I guess?
Yeah, so that's a pretty general Peaks question. So basically the question is, you have a different set of PTMs for de novo sequencing and Peaks DB, which is possible. So going a little bit deeper into the algorithm, so Peaks generates a protein shortlist based on the de novo sequencing results, and also considers the de novo sequencing result when scoring the result. So if you have a different set of PTMs for Peaks DB and de novo sequencing, if let's say Peaks DB finds an extra PTM that de novo sequencing missed, it would be a slight difference at that position in terms of comparing the de novo sequencing result to the Peaks DB result. So it would be a slight difference in the score, but the similarity in the sequence across the rest of the peptide should be quite similar. So for most peptides, it won't change whether or not the peptide passes or fails the false discovery rate. So it should be, it should have a minor effect, but it shouldn't have a huge effect on your results.
Hey, great. And there's just a last minute question here. It says, I'm a beginner to Peak Studio software, and because of your demo, we've seen that you can do area coverage. Is it possible to see in Peak Studio the area coverage of a protein as well?
What do you mean in Peaks Online?
I think in the demo they saw it in Peaks Online, and they're just wondering if it's in Peak Studio as well.
Area coverage. Oh yeah, for sure. Yeah, that's the exact same between Peak Studio and Online. Yeah.
And if anyone does have any questions specifically with like the similarities between the two, we can always set up a trial for you to give it a go and see how the results do look. And Dan and the application team would be happy to walk through the demos with you as well.
Excuse me, may I still ask a question?
Sure.
Oh, because it's a user meeting, so, you know, I have an issue here on the PTM spectral file from PTM search as compared with DB search. We just chat online with Juan Rojas. So the question it is why we cannot export the spectral libraries from PICS and only from PICS-DB search. Would be very helpful. I use other software, and having the PTM in the spectral library really improved the coverage of the immunopeptidomes and proteomes. Everything is better if you can have a spectral libraries with PTM based on sample. Yes, so.
Yeah, I can answer that. Well, so currently you can only create spectral libraries from a PEAKS DB result, not from a PEAKS PTM result. But for any PTMs that you include in a base PEAKS DB search, it will still be able to create a library spectrum from your modified peptide. So it becomes a bit of a problem if you have 20 PTMs, then it's not really then it might take a long time to finish that search, but you're still going to be able to have those 20 PTMs in your library. When you start to get too many PTMs, then it starts to become less reliable when you then use those many different possible PTMs in your library, and then starts to create problems for the following library search. So at the moment, we're just avoiding any problems there.
Yeah, I got it. So in other words, you want to make the DB search with a targeted PTM, and then you export. When you export the spectral library, the PTM are exported. Great. Okay.
Yeah, exactly. Yeah.
Thank you. Great talk.
Yeah, thanks.
Great. Thanks again, Dan. And then moving on. So Zia, if you want to go ahead and share your screen. Our next section is actually going to be a two-part presentation of the emerging research and applications that BSI is involved in. First, we're going to be hearing from Zia Rahman, who, like Dan, has been a longtime member of the PEAKS team. And Zia obtained his master's of mathematics and computer science from the University of Waterloo in 2007. And in that same year, he joined the bioinformatics solutions as well. Since then, he has played a very important role to where the product is today, from his knowledge of handling the various vendor data formats and the relationships with all the different vendors, and to leading the PEAKS development team as the product manager since 2015. And then in 2018, Zia was promoted to VP of production. And so today, Zia is going to be talking about a new tool that the team has been working on.
Hello, everyone. So is my screen shared?
Yes, it is.
Okay. So hello again. So today I'm going to introduce you to the new software tool that we are working around. So is the GlycanFinder, our solution for the glycoproteomics. So before going into the software, first let's start some basics. Just give me a second. Okay, so the basic building blocks for the glycoproteomics is the monosaccharides, which are just the simple sugars, glucose, which is the most common of them. When these monosaccharides binds with other monosaccharides, they form the polysaccharides, which are known as glycans. And when these glycans ultimately binds with protein or some other biological molecules, that by a chemical process, then we get our, that process is called glycosylation, and then we get the glycosylated proteins. So this is one of the most common post-translational modification. And the glycoproteomics is the study of this glycosylated proteins. So basically, we want to know what are the glycosylation sites, which glycans are involved in that site, and is there any ways to quantify them? So recently, glycoproteomics is getting more and more attention. Some recent developments in instruments helps us to develop high throughput workflows, most tailored towards the identification of glycans. And also at the same time, the bioinformatics section also came up with some innovative ideas about how to identify the glycopeptides and their binding sites. So as a result, we have seen enormous development in this area. Our GlycanFinder is our solution for the glycoproteomics. So we basically try to find solutions for two types of glycosylation and linked glycosylation, where the glycan binds to the nitrogen of asparagine. These glycans usually have some core structures, fixed core structures, and the binding site also has a motif. And this type of glycosylation is more prevalent than others. And the other glycosylation type that we also handle is the O-glycan. This type of glycans, they attach to the oxygen of serine or threonine. So let's look at the basic workflow in the GlycanFinder. So we start with the LC-MS data. The data goes through a spectra classifier first. So we classify the MS2 spectrum into glycospectra and non-glycospectra. From MS1, we do the feature detection, and these features will be used later for the glycan profiling. For the non-glycospectra, those are passed to our DB search engine, and our DB search will provide us a native peptides list and a candidate protein list. This candidate protein list is passed on to the GlycanFinder search engine. So the GlycanFinder search engine, along with this candidate protein list, it will also use a built-in glycan database. So, and then it will do the searching on the glycospectra, and ultimately it will come up with glycopeptides and glycoproteins from the search. So finally, in the software, we put together these features, glycopeptides, glycoproteins all together, and present it nicely as like in Pixel Studio results we show them. So let's have a look at the glycospectrum. So this is a typical glycospectrum. It has some characteristics which are different from a typical peptide spectrum, that MS2 spectrum that we see in the proteomics data. So in the lower mass region, we can see some oxonium ions, which are signatures that this spectrum can be used for glycopeptide identification. And at the higher mass range, around 2000 Dalton or more, we can see a series of high intensity peaks over there. So those are the glycopeptide peaks. Basically, they give us some ideas about glycan candidates. So from this information, along with the precursor, we can actually come up with a candidate list for glycans. And from then we look at the lower mass region and find those peptide fragment ions and the glycan fragment ions to find the peptides. Obviously, there will be multiple candidates for glycan and also the peptides. And then our algorithm comes up with a scoring function that considers so many factors and ultimately came up with the best match for this spectrum. So this is the basic way how it works. So now let's look at the software. So before going into the GlycanFinder tools, I will just take a few seconds to tell you that earlier Dan demoed us Pix Online, which is our high throughput solution for the distributed environment. It's very highly scalable. It comes with many improvements. The algorithms are optimized for multi-processing. The fast processing workflow is optimized, and we reduce the memory footprint. So the list just goes on, all those improvements. So those are necessary for the distributed environment. And so, but a lot of our users are still using a desktop environment. So we thought that why not incorporate all these good features from into our desktop environment. That leads to the new Pix platform. So we are working on it right now, and our new software tools will be based on this new Pix platform, which will share common core algorithms with Pix Online that make sure that the results will be the same on both environments. So our GlycanFinder is based on this new platform, but in terms of user interface, we try to keep it same as Pix Studio. So there will be no surprise. So this is the main window for GlycanFinder. As you can see, it's pretty similar as in Studio. So we have the taskbar with the main functions on the top. We have the project tree and information panel to the left and the result details on the right-hand side. The first differences that you will notice here is the project structure. So the project structure is different. It's more like it's online now. So projects are now a container of, in the container we have the data. The data can be organized into samples and fractions. And then we perform the analysis on the data. We can select all of the data, or we can select some parts of the data to run a new analysis. The analyses are also containers of the result components. So for example, in the screenshot that you can see now, we have for the GlycanFinder, we have data refinement, GlycanFinder and export. Those are the result components. I will come into a little bit more details about those. Creating projects are simple. Like before, we have the new project button. We click on it, or we select from the file drop-down menu, and the project wizard will pop up. Data selection is exactly the same as in Studio. We select the data and organize them into samples. Then we move on to the workflow selection. Currently, Pix GlycanFinder offers 3 workflows: N-link GlycanSearch, O-link GlycanSearch, and we have one more, which we perform the N-link GlycanSearch first, and then we perform the O-link search on the unidentified spectrum. We have option to save the workflow so that the workflow and the parameters in each steps can be reused for our other projects later. Parameter dialogues is also a dialogue looks similar as a database search dialog. So we have the mass tolerances, enzyme and digestion mode selection. We can specify the modifications, fixed or variable. So these are the non-glycan modifications. All the glycans are also included automatically. And for the identification, we also provide the FASTA database for proteins. And finally, we have some report filters. These are similar to Pix Online report filters. So we provide the report filters in the parameters now, and this will filter and the result will be ready. So when the search is finished, a new analysis node will be generated in the project tree. And if you expand the analysis node, you can see the result components. So the GlycanFinder search results will be in the GlycanFinder node. Just double click on it and you can see the results on the right-hand side. So the results are organized in a similar way as our PixDB result or PixPTM results in Studio. So that if you are familiar with Pix Studio already, you will have no trouble in browsing different areas of the results. So I will not go into details of everything here. So I will just highlight a few important things. So the all the glycopeptides are put in the glycan tab. So the table contains the peptide and information and also the glycan information. For the selected peptide, we show the best PSF by default as an annotated spectrum in the spectrum view. So the annotation shows us the glycan structure, and the fragment ions are annotated with the corresponding glycan fragments. We have the ion match table that shows the glycan fragments, so their theoretical and observed and Z values. And also we have some little bit of profile information about that peptide. I will come to the profiling in more details later. So So if our experiment involved replicates, or we have multiple samples in the analysis involved, then we automatically perform the match between runs. So not only identification, after that we do the match between run between those different runs to align the identified features together. And this aligned match between run results can be observed in the feature table. So we have the peptide table. We can, in the profile columns, will give us the area for those peptides. And then we can see the details in the aligned features and aligned retention time alignment, and also the XICs. And the glycoproteins are grouped in the proteins table. So in the proteins table, again, we have the protein information, their coverage score, and along with which glycans are found, and the glycosylation sites are listed in that protein. And the coverage pane, we have more details about the glycopeptide coverage and also the native protein peptide coverage there. So the glycopeptides are highlighted here with purple color, also in the coverage bar in the coverage pane below. Glycosylation sites are also marked with the purple color of the residue. So in this screenshot, there are 2 glycosylation sites that we can see. Now I will spend some time on the glycan profiling part. So if we are interested on a particular glycosylation site, then we can see the glycan profiling for that site. Pretty simple, just click on that glycosylation sites in the coverage pane, and a dialog will pop up with the profile information. So the table will show us all the glycans we found there, and their moiety, their area, proportional areas, and we have the match between run information is also there. So how much we found in different samples. And this information we can also see as a pie chart. And if we are interested on profiling for all of the glycan site together, so there is another tool we have, is the glycan profiler tool, which is available at the toolbar of the coverage pane. So we just simply click that, and the glycan profiler will be displayed its results in a dialog. So in the table, you can see that all the glycan sites are listed there with the corresponding glycan found and the area for that peptide. And also we have those information represented in different types of charts. So we have a bar chart for each positions. And also we have separate XIC charts for each of these position and moiety separately. We have a few others, statistical information in other places. One is the glycosylation site summarization table. So in the summary page, we can see this. So this summary table contains only the top 20 glycans. So this summarizes our findings and the total analysis. This table can be exported, and the exporting will actually contain not only 20, it will contain the complete list. Now let's come to the exporting. So the exporting results is available from the exporting node under the analysis. So currently we have a bunch of text export in the comma-separated versions. So we select all those whatever option we need actually for our research. So we can select those options. We can select the destination directory, and then just click the export button. Those text files will be saved, and they can be opened in any Excel or other spreadsheet, and they will be ready for further analysis. So basically, this is our software GlycanFinder. So it's a PIX tool and built on the PIX platform. And we have the similar user interfaces so that we don't see, it will be very easy to operate if you are already familiar with PIX Studio. Those who are interested, we have ASMS poster relevant. It's a virtual poster, so we put some introduction and findings in this poster. So yeah, that's it for our new software tools. So these tools will be ready for beta release pretty soon. So then our users will be able to have a first-hand experience on it. Catherine, yep.
Great, thank you, Zia. So there is a question here. It says, does the GlycanFinder provide pie chart abundance of different glycans at the same site? And can you use GlycanFinder to quantify different glycan changes at the same site between different samples as well?
Yes, that's right. So the pie chart shows us the abundance of different glycans at that same site.
And even between different samples?
Yes, for different samples, we have to go into the profiler. It is more details profiling tool.
Okay, perfect.
Yeah.
Can I have
yes.
Thank you. Great talk. So for the users who want to access the GlycanFinder, so we have the licenses. We need to buy a new license for GlycanFinder. That's one question.
Catherine, I think it's for you.
Oh.
So with the GlycanFinder, it's going to be built into the new PIX Studio platform. So we're not quite sure how licensing is going to be distributed yet, but when that time comes, I'll make sure to update you.
Okay, and another question for Dr. Rahman. Thank you for your great talk. So were these data you presented from proteins enriched at the level of the proteome, the glycoproteome, or for the proteins enriched at the level of peptides, glycopeptides? Which one gives you the best coverage?
Okay, so let me answer this way. So our algorithm can handle. So you mean the enriched glyco?
Yeah, I mean from proteomic data.
Yes. So our workflows.
Yeah.
Yes, our workflows can handle actually both. So obviously, if it is glycoproteomics data, then you will get the better result. You will get more confident identification for glycans there. But for the general proteomics data also can be used, and we can find some results from there. So internally, our workflows are actually will be a little bit different, but from the user's perspective, you will see no difference in there.
Thank you.
Great. Okay. Well, thank you again, Zia, for that presentation. And we're just going to move on since we're quite over time. But for the second part of the session, we're going to be hearing from Dr. Kyle Hoffman, who is a senior application scientist at Bioinformatics Solutions. He completed a master's in science in cell and molecular biology in 2012 at Western University, and also followed by a PhD in biochemistry in 2017 at Western as well. Shortly after that, Dr. Hoffman took on a postdoctoral associate position at Yale University with Professor Dieter Suhr in molecular biophysics and biochemistry department, where he performed research in the synthetic biology field. Upon returning back to Canada, he then began working at BSI in 2019 and is currently focused on antibody sequencing and immunopeptidomics. So over to you, Kyle.
Thanks, Catherine. I apologize if some of this is going to be a little bit repetitive. I know we've already heard a couple of great talks in the immunopeptidomics field. But for my talk, I just wanted to highlight the importance of doing de novo sequencing and how peaks de novo sequencing can uncover a lot of antigens, HLA peptides that will not otherwise be identified in a generic database search. Okay, so as we've heard, the study of immunopeptidomics involves identifying these immunogenic peptides displayed on the cell surface by HLA complexes. And I just want to briefly go over the processing pathways and how the peptides are derived from proteins in order to be displayed on the cell surface, because it's very important for their identification. And so, as we've heard, a lot of the antigens that are being displayed aren't just from your typical protein coding genes, and therefore they may not be identified in a standard database search such as Uniprot. And so for these reasons, there are many different complex processing pathways that can lead to these HLA peptides being sort of imported into the ER for assembly into these complexes. But these sequence modifications can arise at the DNA level, so we can have genetic variants, but also at the RNA level, things like RNA editing and transcript splicing can lead to different peptide sequences. And at the translational level, we now know from ribosome profiling data, as well as some RNA-seq data, that the HLA peptides can be derived from non-coding regions, alternative open reading frames. So we've seen peptides that have been derived from translation that starts or initiates from codons other than AUG, for example, even codon read-through, and also from expression or translation of transposable elements, and also defective ribosomal products. And so after translation, proteins will get degraded by the proteasome and sort of chewed up into shorter peptides that are then transported into the ER through the TAP complex for assembly into HLA complexes, and then transported to the cell surface for display. And so, yeah, since many of these cannot be identified in generic databases, that's where sort of the power of de novo sequencing comes in to help identify these other products. And so here's the workflow. It's pretty straightforward, and it's very similar to the one that Andy had shown earlier. So basically, you start from purified HLA peptides in a raw mass spectrometry data set. It first undergoes de novo sequencing, and then we perform a database search, but also collect all of the or keep all of the peptides that were identified by de novo sequencing only and don't match anything in the database. And then for both groups, we perform a spider search to identify mutated peptides in each group. And then any peptides that pass through these filters shown in the middle, and also meet the criteria of being a predicted HLA ligand, will get grouped into the HLA-1 ligandome. And so I've also used the NET MHC PAN tool in order to predict binding affinity of HLA peptides. So all of the ones that I'll show that are predicted as HLA binders have scored less than a 2% rank in the eluted ligand data. So I chose to sort of demonstrate the utility of this workflow. I chose to reanalyze the data that came out of this paper by Albert Heck and Wei Wu. And so in this paper, they performed a standard database search with their data against the human SwissProt database. And so this paper involves samples or biopsies taken from a single patient. And so they took tissue biopsies from both normal colon tissue as well as a colorectal cancer tumor. They derived 4 distinct clones from the tumor and expanded these into organoids, as well as expanding the normal healthy colon tissue into an organoid, and then purified out the HLA class 1 complexes and eluted the bound peptides for MS analysis. And so all the peptides that they identified in their database search were then compared to, between tumor samples and normal samples, they were compared. And what they found is that there was a large overlap between each of the tumors with peptides identified in normal tissue. However, they then go on to show that although there's a small fraction that are unique to each of the tumors, they are fairly abundant. And so in panel B here, they show from tumor clone 1, they ranked these based on decreasing intensity, and the blue dots shown along this curve represent peptides unique to tumor clone 1. And so you can see that although they represent a small portion of the total tumor clone ligandome, they are fairly abundant on this curve. And in panel C, they show that they did the binding affinity rank using net MHC pan as well, and rank them based on their peptide binding rank. And as you can see, the small sort of white dots along this curve represent peptides unique to tumor clones. And so anything in this sort of green area is predicted as a strong binder to HLA proteins. In the middle here are weak binders and non-binders at the top in the pink area. And so again, they do point out that a number of the peptides unique to tumor clones do score fairly well and are predicted as strong binders amongst the data. And then they characterize the overall length of the ligandome sets in each of the samples. So as expected, most of the peptides are 9 mers, which is very characteristic of HLA-1 peptides. And they do point out that they see a slight increase in longer peptides, 11 and 12 mer peptides, amongst those that are unique to tumor clones. And so I ran this, the raw data through peaks, and these are the parameters that I use. Basically, they reflect what was used in their paper. They ran their samples in triplicate and used both HCD and ETHCD fragmentation mode, and searched it against the human Swiss UniProt database, filtered out any contaminants. I chose to use a fairly stringent ALC score to include only high quality de novo only peptides, filtered by length, 3% FDR, and removed any duplicates. So here are the results from each of the tumor clones as well as normal tissue. You can see there's quite a significant amount of peptides that fall into the de novo only category. And on average, about 70% of these were predicted as HLA binders. And in comparison, here are the number of peptides for each sample that were identified in the database search, with a slightly higher percent of binding prediction, around 90%. And on the right side here, as you can see, of those peptides predicted to bind HLA proteins, over 50% of them were not identified in the database search and fall into the de novo only category. So further emphasizing the fact that de novo sequencing is necessary in order to further uncover such peptides. I then did a comparison between peptides identified in across all tumor samples and normal tissue, and I found actually quite different result, where there's only about 36% of them that are found common to both. But what was interesting is when you break this down into peptides identified in database search versus de novo only sequence peptides, you can see that over 53% of them overlap from the database identified peptides, whereas only about 26% are found common in the de novo only sequence peptides. And this paper sort of focused on the the heterogeneity that can be observed amongst clones from the same tumor. And so I decided to go ahead and do a similar type of analysis here. And so amongst all the 4 tumor clones, this is combined de novo only peptides as well as database identified peptides. There's about a 26% of the peptides are found common to all clones. But as you can see, there's quite a few, between 1,000 and over 2,000 peptides that can be unique to any in particular clone. But again, when you look at the database identified peptides, we see sort of a different picture. So 45% are common across all peptides, whereas in the de novo only result, we see only 15% that are found common across all clones. And so this is, again, driving home an important point that de novo sequencing will uncover a larger diversity among peptides, across different clones. And I characterize the peptide length from peptides in normal tissue, those across all tumors, and then unique to tumors. And what was consistent was there was a slight increase in 12 mers or longer peptides found in those unique to tumors, which was similar to what they found in the study. I then went on to use the mixed MHC-P tool to generate sequence motifs for each of the HLA alleles expressed in this patient sample. And so from the tumor-only peptides fit nicely to each of the sequence motifs for these respective alleles. And for the most part, they were fairly similar to the sequence motifs generated from peptides from normal tissue, although I did notice a few differences between the two, such as at position 9 in the HLE B motifs, there was a stronger preference for tyrosine at position 9, whereas that seemed to be lost in peptides that were specific to tumor samples. And maybe a slight increase in proline represented at position 4 in normal tissue and less pronounced in tumor samples. And lastly, I wanted to highlight one of the peptides. We found a mutated peptide that was specific to the tumor samples. And this peptide wasn't initially identified in a database search. So we found it through in the de novo only list of peptides. And then when performing a spider search that allows for mismatches in the database search, we identified this valine to alanine mutation at position 29 in a cyclin-dependent kinase. And interestingly enough, this is a protein that is known to play an important role in cancer progression in colorectal cancer tumors. So in summary, Peak's Novo sequencing captures a larger diversity of HLA-1 ligands compared to database searching. And there's a significant increase in the number of ligands identified, fairly high accuracy. So I predicted it to be so of the peptides identified, 70 to 90% had were predicted to bind HLA proteins. And there are differences in sequence motifs that can be identified between tumor and normal tissue samples. And de novo sequencing coupled with the spider algorithm could potentially identify neoantigens in the data. So obviously there's more experiments that need to be done to further define this as a neoantigen. And I didn't perform any immunogenicity assays here, but it was interesting that you can uncover these mutated peptides in the de novo data that would otherwise be missed in a standard database search. Okay, and that is it. And I'll take any questions.
Great, thank you, Kyle. Does anyone have any questions?
Can I ask a question? So, hey, Kyle, great talk. So I just wondering, when they do these tumor versus normal, so are they using DDA or DIA? How many replicates have been performed? Because in my experience, actually, the the replicate, even replicates between the immunopeptidome is not great as like tryptic peptides. So just the reason I've been stated before, like probably the sample prep is really challenged for this for the pipeline. So have you looked into their details of their data?
Yeah, it's all DDA data, and each sample was done in triplicate. Okay. So they eluted from 3 replicates for each sample. But yeah, I'm not sure how reproducible like the peptide identification was across those triplicates. It was sort of just, they're all just sort of grouped together in peptide identification.
And the missing value is one of the major issue actually known to be for DDA, right? So that's why like even our lab actually using DIA a lot. Like we basically not using DDA that much as before. But you know, I mean that is really a like issue with the DDA, I think. Yeah.
Yeah.
Okay, and if there's any other questions, please feel free to reach out to the Peaks team. Kyle, if it's okay, I'm gonna move on to Tony's talk just because we're way over time here. Our final presentation, and we've been very excited for this talk. As Paul mentioned, Tony is actually one of the members of BSI Scientific Advisory Board. So Tony is an NHMRC Principal Research Fellow and Deputy Head of Research of the Department of Biochemistry at Monash University. He's also the Vice President of the Australasian Proteomics Society and a member of the HUPO Council. He is also currently a visiting professor in cancer science at the University of Southampton. And his laboratory focuses on how the immunopeptidome is influenced by infection, inflammation, and the environment. He has made important contributions to understanding the role of antigen presentation, including the characterizing peptide epitopes involved in T-cell recognition in several autoimmune diseases, drug hypersensitivity, cancer, and infectious diseases. He has really been instrumental in bringing new quantitative tools to immunological studies, and in particular, the use of multiple reaction monitoring to calculate the cell surface expression levels of specific HeLa peptide complexes. His lab is one of the first laboratories to adopt new data-independent acquisition techniques and apply it to quantitate antigen presentation at the protein and peptide epitope level, as well as understanding complex host pathogen interactions in infectious and parasitic diseases. As one of the most cited Australian proteomics researchers, Tony really is a leader in the field of immunopeptidomics with over 200 related publications. And so Tony, whenever you're ready.
Well, hopefully I live up to your introduction, Catherine. So hopefully you can see my screen and hear me clearly.
Yes, we can.
Well, it's morning here in Melbourne, and we've had a really stormy evening. So around about 5 o'clock this morning, I decided to drive into work and dodge trees and branches to make sure I had a stable internet connection. So I might look a little rough around the edges, and I might sound a little rough around the edges too. I should really thank everyone for hanging on for this last talk. I know we've gone over time, and I think I'll try and save a bit of time because we've heard some excellent introduction to what immunopeptidomics is. And it's really gratifying to see just how many talks on immunopeptidomics there are in this Peaks user group meeting. And, you know, I think that's probably a reflection of, I think, many people's preferences to use Peaks as a search tool for this type of data. And particularly today, what I'd like to do is talk about the dark immunopeptidome. So I'll define what that is in a minute. But first, I thought I'd better do some disclosures. So as Catherine's just told you, I'm the scientific advisor for Bioinformatics Solutions. And obviously that's the main disclosure I need to make for this talk. I'm on the scientific advisory board of Vaccine Biotech, who use AI to design vaccines and heavily use immunopeptidomics data in that respect. Also had collaborative research agreements with Bruker, Daltonics and SciEx, and I'll be showing you some data from 2 relatively new platforms from both of those vendors today. And we get a little bit of funding from a few companies which isn't particularly relevant for the talk today. So I'm going to sort of absolutely rocket through this slide. We've all heard that T cells recognize peptides that decorate the cell surface in complex with either MHC class 1 molecules or HLA class 1 molecules in humans, or MHC class 2 molecules. And you've heard from Cristina and from Andy and Kyle about both these pathways. So I won't dwell on it too much longer. But what I would like to do is just pause a moment and think about, what this immunopeptidome is. This collection of peptides that end up bound to HLA molecules and presented on the cell surface essentially for immune surveillance. So for T cells to look at a snapshot of the cellular proteome and ask, is something different, and should an immune response be elicited? Obviously, cells don't ask these questions, and I probably shouldn't personify them too much, but essentially that's the deal. So we know that the immunopeptidome is heavily influenced by the presence of pathogens or during some sort of malignant transformation, and we've heard from Andy, for example, about that already. The composition of the immunopeptidome is highly dependent on the background genetics of the individual. And this is probably one of the biggest challenges, particularly for clinical and human immunopeptidomics, that we all express 6 different HLA alleles, typically of the class 1 flavor, at least 6 of the class 2 flavor, and each of us will have different immunogenetics. So we will all select a different snapshot of that cellular proteome to display to our T cells. We know that the immunopeptidome is influenced by the cellular microenvironment, and I'll talk a little bit about how pro-inflammatory environments can mold the immunopeptidome. And it contains novel sources of antigen, some more contentious than others, I will admit. And this really is the dark immunopeptidome. So my definition of the dark immunopeptidome is the approximately 40% of high quality MSMS spectra we get during immunopeptidomics acquisition that we cannot match to a reference proteome. And we know that this can contain things such as endogenous retroviral elements, sort of ancient viruses that have integrated into the human genome and can express antigens that can be presented. Different forms of novel open reading frames, so gene fusions, untranslated regions, alternative reading frames, antisense encoded reading frames, non-coding RNA, which obviously does code for some things, including antigens. And most recently, in infectious disease, a very interesting paper published in Cell last year, where they identified during viral infection these so-called untranslated Frankensteinian open reading frames, or UFOs, which is just a really cool name, so I thought I would include it. Also, post-translationally modified peptides, and for those of you in the field, you'll know that we've studied quite extensively proteasome catalyzed splice peptides. Some might argue to our detriment, but we're still quite strong advocates for those, and I'll explain a little about those momentarily. And also, we've become very interested, particularly in clinical immunopeptidomics applications, in terms of looking at environmental and microbiome-derived peptide antigens, which have important implications for recognition of various tissues. There's a very nice paper published in Nature recently identifying bacterial peptides. So there's a bacteria that colonize a tumor, a melanoma in this case, and showing that they can be presented to the T cells and potentially have some sort of immunomodulatory effect. We've worked quite extensively with gut biopsies, for example, where we're interested both in food-based antigens and also the microbiome and how that contributes to the sorts of peptides that are presented. And I'll make arguments in some cases that nearly for all these sources of the dark immunopeptidome, using de novo-based sequencing is by far the preferred way to identify these sort of peptides. So many of you are probably used to proteomics, and those workflows are relatively well established. There are many challenges in immunopeptidomics type of analyses that really differentiate the way we work with these samples. So typically these are highly complex mixtures, and they're complex mixtures of very similar peptides. So you saw in Kyle's talk that different HLA molecules have a preference for certain sequence features of the peptides. We call these anchor residues. These are residues of the bound peptide ligand that go down into the cleft of the HLA molecule and are involved in binding interactions. So they're somewhat conserved within the immunopeptidome of a certain immunogenetic background. They also typically tend to be, for class 1 peptides, have a very small length distribution. So typically over 80% of the peptides are anonymous. So again, you've got very similar peptides, all very similar length, don't necessarily separate particularly well, and present quite a challenge to analyze using compared to more standard proteomic workflows. So similar length, large dynamic range of concentrations, anywhere from 10,000 copies down to 1 copy of a given peptide antigen bound to a HLA molecule per cell. And we know that T cells are really clever and super sensitive. And even down at this level, we know this can be actually physiologically significant. So we need to be able to identify the abundant peptides and drill down into the low abundance peptides. And as I mentioned, there's a potential for many isobaric species. These endogenous peptides are not predictable. So these are not tryptic peptides. These are generated through, as you've seen, quite a complex mechanism involving the proteasome and other peptidases that trim these peptides in a relatively unpredictable way. And being non-tryptic peptides, they don't always give us the nicest B and Y ion set that allow us to get very confident sequencing. They're also often singly charged peptides, and as you know, we often avoid these peptides because of the chemical background in the sample, and we can miss out on up to around 20% of the bound ligands that we can at least detect at the moment. And of course, we still have this issue with isoleucine-leucine discrimination. This is a common amino acid, and it's commonly used by some of the HLA molecules as an anchor residue. So we often get some ambiguity in the identifications. And probably the biggest challenge that we face is that every ID we make is a singleton. We don't have that nice corroborative evidence of seeing 10 tryptic peptides from our favorite protein to identify the protein. Every peptide antigen is identified based on the quality of the spectra. And this is where de novo sequencing in particular is absolutely key. And I would argue that these first 2 points here can be addressed by technology, and I'll show you a little bit about how we can do that. The third, obviously, with the informatics is what we're dealing with pigs. So potentially constraining the potentially astronomical search space to allow us to get confident identification and to not have false discovery totally hamper our analysis. And to deal with these non-canonical antigen sources, which often aren't templated in the genome and not easily inferred. So this essentially recapitulates what I've just said, and for time, I'll just skip this slide. And what I'd like to do today is talk about the dark immunopeptidome in the context of cancer. So you've all sort of seen this workflow already. We work with cells, patient-derived cell lines. We work with tissues, biopsies, etc. And the basic protocol is the same. We lyse the cells in neonative conditions. We use a variety of different monoclonal antibodies to capture these HLA peptide complexes. We then acidify the bound complexes are mobilized on this affinity matrix. And what this does is facilitate the elution of the peptide and dissociation of the peptide away from the MHC proteinaceous components. We, in particular, when we have enough material, use reverse phase HPLC to fractionate the peptides to get at the complexity of the immunopeptidome. And we analyze the various peptide fractions by high resolution mass spectrometry. And typically in a run, we'll be working with somewhere between 10 and 60 concatenated fractions. So we put a lot of effort and time and instrument time in these sorts of analyses. We do our database search, and we've been talking about this already. And then we analyze the peptide characteristics and validate the peptides. And this is another key step for peptides that are actionable. For example, a neoepitope, we would synthesize that synthetic peptide and show that it has identical chromatographic, ion mobility, MSMS fragmentation characteristics as the candidate that we think we've identified. And obviously, we've published quite extensively our methodology. The one in Nature Protocols, highly cited paper with the methodology for this sort of large-scale application, and more recently, a paper in Current Protocols where we've sort of miniaturized this isolation for scarcer samples. So what is our strategy, and how do we go about cancer epitope discovery? So we take our tumor tissue, cell lines, etc. We'll often treat the cell lines or tumors with under different conditions. And one we like to use a lot is interferon gamma treatment. And we've published a few times already to show that interferon gamma can really remold the immunopeptidome. And so by having these different conditions, we expand the potential immunopeptidome of these cells. We combine, as you heard from Andy, sort of genomic information about the tumor to generate a bespoke database. And we combine that with our LC-MS data. We obviously use peaks, otherwise I wouldn't be here discussing this with you. And we really do leverage the de novo assisted or de novo only sequencing to get at the peptides that we can't assign to a reference proteome. And in doing so, we were able to identify de novo neoepitope candidates, and you've heard how this can work already from Kyle. And often we were able to map this back to the whole genome sequence or RNA-seq data. We can look at non-canonical antigens, and we'll discuss these in more detail. And we're also able to access these post-translationally spliced peptide antigens, and I'll talk a little bit about that in the context of colorectal cancer in a slide or two. We're also interested in other PTMs, so not just these spliced peptide antigens, but a whole series of cancer-specific PTMs. Andy showed you some of these already. And I guess some of our favorites include deamidated peptides and also phospholipeptides, which can be a little harder to find because of their, I guess, resistance to ionize well. So using these sorts of approaches, what can you expect to see? And this is a question I get a lot, so I thought I'd put this slide up. So if we're using a cell line, we look maybe at a couple of different culture conditions. We would anticipate we'd be able to see between 50 and 100,000 unique peptides from that particular cancer cell. If we're working with more scarce amounts of tissue or with cancer biopsies, around 100 milligrams or more, we get about a fifth less in terms of unique peptide identifications. We also work in DIA, but today I'm simply going to talk about DDA for time and simplicity. But we do use spectral libraries for confirmation and for workflows such as MRMHR. And we're very interested in applying AI-based approaches, and obviously a lot of this can happen within the Peak software to improve our peptide matches, because we are using this very data-centric, peptide-centric workflow. So retention time predictors, not my personal favorite, as you may already have heard. MSMS predictors, or the use of extensive libraries based on a large database we have of identified peptides. We've been particularly interested in looking at predicting these collisional cross-sectional areas from data, particularly from the Bruker-Timstorff. And obviously de novo sequencing and the work with the guys at BSI. What we do, and what I will say, is that we're interested in all classes of tumor antigens. So cancer testis antigens, neoepitopes, various types of PTMs, including spliced peptides. And I think the reason for that should be evident from the little snippet I'll show from some colon cancer work. So this is unpublished work, but I think it sort of highlights quite nicely why you need to have a fairly broad sort of view of what a cancer epitope might look like. So what the data I'll show you is sort of highlighting two different phenotypes of colon cancer, so-called microsatellite stable colon cancer, which has low mutational burden and it has sufficient DNA repair, through to a microsatellite unstable phenotype, which, because it does not have DNA repair, has increased mutational burden. And, you know, somewhat paradoxically, these unstable phenotypes, which sound worse, actually aren't so bad if you're the patient, because this high mutational burden leads to improved patient outcomes, the infiltration of the tumors by tumor infiltrating lymphocytes, and the subsequent responsiveness to checkpoint inhibition. So I'm going to show two snippets of data. I'm not going to go into huge details of the data. You've heard and you've seen this type of data, and I just don't think I've got time to go into great lot of detail. So in the first example, I'm showing you data from two very well-studied, well-characterized patient-derived cell lines. The interesting thing about these two cell lines is that they are derived from the same patient. One is derived from the primary tumor. The second derived, I think, around 12 months later from a metastatic tumor isolated from, I think, a lymph node. So SW480 is the primary, SW620 is the metastatic derived cell line. And when we analyze these cells across these two different conditions of plus and minus interferon gamma, and we combine all the data together and stick it on one slide, what we're able to show is that we can identify around about 32,000 peptides at a 1% false discovery rate across these two conditions. So not very high expression of class 1 on these cell lines. Now, everyone is interested in identifying neopeptides, so these peptides that contain mutations. And based on genomics and a sort of filtering approach where we filtered out the potential binders, we looked at around about 1,500 possible neoepitopes that could be present in the immunopeptidome of this particular cell line. And despite looking really hard with this extensive fractionation, spending far too much time on the instrument, we're only really able to confidently identify one mutated peptide from all these possibilities. What we do find, though, is other rich sources of potentially actionable peptides. So we're able to show that of the PTM peptides, around 12.5% could be annotated as spliced peptides. And I know there are other potential explanations for these, but the one thing I do feel with these spliced peptides is however they're generated, the de novo sequencing gives us high confidence that we have the sequence right. You know, mass specs never lie. This is a mantra that I tell all my students. Mass specs never lie. We just often misinterpret the data. But around 12.5% of these peptides aren't templated to the genome and are potentially post-translationally spliced. And this is by far the highest category of PTMs, with around about 1.5% of the rest of the PTMs dominated by either deamidated or phosphorylated peptides. The other point is that around 2,000 of these peptides are derived potentially from cancer-associated antigens. And the big question comes then when you see so many is, how do you stratify or prioritize these for any sort of clinical studies? If we move now to this microsatellite unstable and MSA high phenotype, and we look at a couple of adenocarcinomas from bowel cancer, again, these are quite well studied cell lines, HCA7, HCT116, throughout the literature used in a great deal. In this study, we actually used two different antibodies to pull down the class 1 molecules. BB7.2, which specifically pulls down HLA-A2, this very common Caucasian HLA allele. And subsequent to that in serial, W632, which is a pan class 1, so pulling down all the other class 1 alleles from the surface of this cell line. Again, what you can see is we get quite good numbers of bound peptides for both cell lines. And because they share HLA molecules, in particular, share HLA-A2, we do see quite a good overlap in identified peptides between these two cell lines. Again, relatively stringent 1% false discovery rate cutoff. And what we observe in these high mutational burden cancers is that now we can really get into the neoepitopes. So again, we see PTMs, again, we see cancer-associated antigens, again, we see splice peptides. But now we're starting to see highly cancer-specific neoepitopes. And what is really interesting, I think, in these MSI high cancers is that we start to see many frameshift mutations. And this is quite characteristic of the genetic defects in these cells. And all these frameshift mutations were mapped back to whole genome sequencing by our collaborators at the Olivia Newton-John Cancer Research Institute. So just to recap, SW480, the primary tumor from this particular patient, only one mutation found. And we had to search quite extensively to find any evidence for a neopeptide in this microsatellite stable colorectal cancer. HCT116, we found 18 mutated peptides, so instantly we're getting more bang for our buck, if you like. And for HCA7, quite a rich source of these mutated peptides, including a number of these frameshift variants, etc. But really excitingly, I think, 4 of these mutated peptides were shared with this other tumor. These are from totally unrelated individuals, yet we're finding 4 neoepitopes that are shared between both. And as Andy sort of alluded to in his talk, this is kind of the holy grail, if you like, that we're able to drill down and find peptides that potentially could be used to treat multiple individuals, all with this shared HLA-A2 allele. So I told you that technology can help, and I thought I'd just sort of highlight something that we've been doing with Brooker on the timsTOF Pro. And this is really accessing the ion mobility, the trapped ion mobility on this instrument. And the point here is that whatever you do, don't throw out the singly charged peptides, because around about 20% of the neoantigens we identify, we only ever see as a singly charged peptide. We don't see it as doubly or triply, and these are the ones that are nicer to work with because they give you better fragmentation. But if your peptide sequence isn't amenable to getting multiple charges onto it, then you have to access them somewhere. And the really nice thing about the timsTOF instrument is that we can sort of use the ion mobility component of the separation we get of these peptides to really focus in on the region of what we call the TIMS plot, where we know these are mostly peptides. Most of this stuff is just junk, polymer, etc. And we can really target the analysis of these in a way that's not really possible on other platforms. The other challenge that we face is this isoleucine-leucine ambiguity. So when we do our de novo sequencing, the big problem is we can't really distinguish between isoleucine and leucine, and generally we just kind of go with one or the other. Then go back to genomes, proteomes, etc., and hope we find a match that makes sense. However, we certainly know that isoleucine and leucine within the sequence of a peptide can be readily differentiated by T cells, so we need to get it right. We need to get the true identity of the peptide as best we can. One of the things we've been doing recently, working with colleagues at SCIEX, who very generously gave us slightly pre-release of this instrument, this new instrument from SCIEX, the 7600 Xenotop instrument, is to ask, well, where can we introduce a different type of fragmentation into our workflows that will allow us to differentiate isoleucine and leucine within these bound peptides. And it just so happens when you use electron-activated dissociation, which generates CNZ ions, quite similar to what you can generate with ETHCD. But the big advantage of this is that you don't need to be working with triply or quadruply charged ions to get decent fragmentation. And we're currently using this in our workflow to see if we can use W ions to get isoleucine, leucine disambiguation, if you like, in our HLA peptide immunopeptidomics workflows. And the short answer to that is, even after just a few weeks, we've been able to start to see disambiguation. And here's an example where both flavors of the peptide are present within a mixture. They are chromatographically separable, obviously. But if you don't believe, like I do, that retention time prediction is that accurate, and here we're looking at a 2-minute difference or so, what you can see for these two peptides here is that we can very easily see either the loss of 29.05 atomic mass units when isoleucine is present, and the corresponding loss of the other side chain fragment when leucine is present. And quite nicely, we can start to use this data, and hopefully we'll be able to use this data using peaks very soon. The last thing I thought I'd just talk about is a different workflow that we've just recently published. And for the sake of time, I think I will just put it this way, that we've been using different databases, using RNA-seq data to generate both a cryptic database of dark immunopeptides, if you like, and a more traditional database search to sort of look to see whether we can drill deeper. And the simple answer is that yes, we can find a number of cryptic peptides using this sort of strategy. And these are the sorts of cryptic peptides we see, and predominantly from these non-coding regions of RNA molecules. So I think quite consistent with what Andy showed. And just to sort of quickly summarize and put everyone out of their misery so we can all go and have a drink or a coffee in my case, because it's too early. So with the sort of workflows we're using, we're getting substantive numbers of neoepitopes when there's a high tumor mutational burden. And I also showed you that when you don't have that high mutation burden, you have to look at other classes of antigens, and we could potentially, if we're all together, we could have an argument about what the best class of those antigens might be. I've shown you that if you're lucky enough to get access to new instruments, the technology can help in terms of both increasing depth of the immunopeptidome and also to get greater confidence in peptide identification. And, the thing that excites me the most about this field, I started doing this in 1994, which is why I've got so much grey hair, is that we're really starting to see the emergence of what I would like to say is clinical immunopeptidomics, and where we'll be able to really translate this into truly personalised approaches for cancer immunotherapy, arguably with quite a broad array of different antigen classes, potentially all in concert to elicit the most efficacious immune response. And so this is just a future perspective slide. I won't dwell on this too much, but I guess the key message is de novo sequencing is becoming increasingly important in these workflows. And we're using it in combination with sample-specific databases provided from various genomic approaches. We're incorporating additional types of fragmentation to improve the quality of the MS-MS spectra that we can generate, in addition to other biophysical properties of these peptides to gain confidence. We're starting to work a lot now with 16S RNA sequencing, metagenomics, and environmental analyses to look at other dark sources of peptides, and in particular dietary antigens and microbiome-derived peptides. And the one thing I would say is that our field has a lot of dirty little secrets, and it's not just immunopeptidomics. I think our experience with genomics is that variant calling is not absolute, and different tools often provide quite a different list of potential neoepitopes. And it'd be great to have a discussion with Andy about this at some point in the future. The peptide-centric nature of our identification needs to be adopted with caution. This is why we always have to validate the actionable peptides using synthetic peptides from my point of view. There's potential for multiple explanations of spectra, some more bizarre than others. And there's even a lack of consensus between the various tools we use to search that data. And I'll just refer you to this very nice paper from my good friend Nicola Tennant and Rob Parker, who show that the choice of search engine really affects both the depth and the peptide repertoires from different HLA class 1 molecules. And perhaps not surprisingly, they showed that peaks managed to outperform most of the other tools that they looked at. And I think that we really need more data-centric approaches to really tap into this enormous variety of antigen sources, particularly in clinical samples. And the challenge really for BSI and other vendors is to how do we maximize confidence in our identification? And are we ever going to get to the point where a list of peptides spat out by a search engine can be taken totally at face value, or are we always going to have to go back and do some form of validation? It just remains to sort of thank a lot of people in my group, and this list certainly isn't exhaustive, so apologies to anyone who's on the line that I haven't mentioned. The work on colon cancer was done in collaboration with David Williams and John Mary Edison at the Olivia Newton-John Cancer Research Institute. They provide a lot of the genomic information that we use to then interrogate the patient-derived cell lines. I've mentioned the collaborations with Bruker and Sykes, and some of the guys we work with there are listed here. We've really enjoyed a fantastic collaboration with BSI, and certainly I'd like to thank Paul and Catherine in particular. Our funding agencies, thank you all for hanging around, and maybe we have time for a couple of questions, Catherine, I'm not sure.
Yeah, no, as long as you're okay for questions. Yeah, if there's any, don't see any in the Q&A. Are there any questions for Tony?
Oh, hi, great talk, like always. I have a question related to your splicing peptides. I know there are many mechanisms proposed for splicing, but specifically they were shown for the insulin-derived peptides in the type 1 or type 2 diabetics. I don't know, for these peptides derived from different cancer antigen or from other sources in the cancer cell line, could you tell us very briefly which are the main splicing mechanism, and do you think that the redox microenvironment, of course the oxidative stress there would affect more splicing, like you have deamidated peptides would have the probability to get better spliced, or I don't know, so I'm just curious.
So I think, when we published our first paper on this back in 2018 now, we were pretty careful to sort of say that we're detecting genomically untemplated peptides, and that we thought the best explanation for them was this mechanism of proteosomal splicing, where instead of getting hydrolysis in the catalytic chamber of the proteasome, under certain circumstances, you get a sort of nucleophilic attack by another peptide fragment, and this ligation of two distinct regions of the peptide together. Now, there's been a huge amount of debate in the literature on this. I think Andy showed a slide from one of the other camp, who I have to say were very selective in the retention time correlations they showed. Look, you can certainly generate these in vitro with purified proteasomes. This can definitely happen. 30 years ago, when I was a peptide chemist, we used to ligate peptide fragments together using trypsin and acetonitrile, because again, if you exclude water, you get these sort of isopeptide bonds formed quite readily.
That is the main mechanism, the proteasome-mediated ligation. That is the major one.
Yeah, so our preferred theory is that there are segments of proteins that just don't digest well in the proteasome, and they kind of constipate the proteasome to some extent. And perhaps this predisposes to the formation of these spliced peptides. They're certainly less abundant than regular linear peptides, if we call them those, but they are found reproducibly in our analyses, And I think there's been some fairly convincing evidence from others that an explanation for some of the sequences we get from this de novo analysis could be based on unusual open reading frames, other untranslated regions as well. But that doesn't explain the bulk of them. So we still have these perfectly good looking MSMS spectra that we can't explain using any other explanation. And I'm quite open to other explanations. It would certainly make our papers easier to get into journals. You know, I think the one thing we do trust are the sequences, because when we test these for T-cell recognition, for example, we get T-cell responses. So, you know, there's a very strong literature from some of the, you know, real leaders in the field of antigen processing, John Udell, you know, Benoit van den Einde and others that show these things can be generated and can be targetable in cancer. The debate really is, I think, around, well, what percentage of the immunopeptidome are spliced peptides? Now, is it 1%? Is it 30%? I don't really care, to be honest, because I still think there's a whole lot in the immunopeptidome we don't even understand where that comes from. So, you know, you can call them alien peptides. You know, they're coming from somewhere, but who knows, right? So, you know, I think rather than really obsess over what proportion they are, I think what the best question is, do they actually matter if you elicit immune response to them? And, you know, I'm sort of hoping that in the next few months, we'll be able to present some data where we show from vaccinated human subjects, we can generate splice peptide specific T cells that kill tumors.
So you were able to have T-cell clone isolated specifically for neoepitope from splice.
So we've used dual tetramer staining to sort out tumor reactive T cells, and you know, hopefully we'll be able to show that pretty soon. We've also knocked out the source antigen of the splice peptides and show that we no longer get recognition. So we can subtract antigen-specific splice peptides from the repertoire as well. So, you know, I think the one thing that, you know, the late great Nilab Shastri said to me was that if you have these crazy ideas, these extraordinary theories, then you need extraordinary proof. And we've spent the last 4 years trying to generate extraordinary proof, rather than, I guess, tip into the arguments that unfortunately pervade our journals. But at least try to avoid it.
Thank you.
So a really, really great talk, Dr. Tony. So what your thought about the PTM peptide? Are they gonna be really a good epitope? Because I was present this work to our clinician at NCI. So to be quite honest, those physicians, the only thing they trust is point mutation, because they know that for years. And then we're trying to propose, for example, the cryptic peptide, fusion protein, you know, PTM. So they're starting to lose interest. When one particular people ask me why their PTM going to be stable. So if this thing can be reversible, changing back and forth like phosphorylation, right? So this we cannot. It's too risky to develop any T cell against this particular epitope. You know, if you infuse to the patient, that's no joking. Like people die. So they, you know, it's just from different perspective. I understand from basic science, we're reporting unbiasedly, mass spec never lies. So, but you know, from clinician perspective, they're only thinking about the safety and the efficacy. So what do you thought about those, like, you know, other than point mutation?
The neoepitope story is by far the most seductive clinically, right? Just as you say, if you have an absolute tumor-specific mutation, then to me that makes sense that you would want to target that. I think things like indels and these frameshift mutations even better, because you're not just got a single point mutation, something that looks really unlike self. So again, potential to not get cross-reactivity. Personally, I think that these other types of epitopes are definitely worthwhile pursuing, maybe in combination with some of the more tumor-specific mutations. But you know, at the moment, we haven't cured cancer, at least as far as I'm aware. So I think, the jury's out in terms of what we should really target. Mutations are great, but maybe not every single cancer cell has that somatic mutation. So, a lot of effort goes into identifying truncal mutations, driver mutations, etc. But we don't know that for every cancer. So I think, for me, if we can detect it on the cell surface, we can't detect it in healthy tissue, etc., then it's as good a candidate as any. Now, with the PTMs, are they stable? Now, we showed quite some time ago that for phospholipeptides, for example, once they're bound to the MHC molecule, when you can cook up recombinant phospholipeptide MHC complexes with a phosphatase, and it's totally stable. So it gets shielded. So, I think the MHC is quite good at stabilizing some PTMs, maybe not all, but for phospholipeptides at least, definitely. And sometimes these phospholipeptides can be a lot more stable than the unmodified version of the peptide as well. So they have a binding advantage. And, the complexity comes from, well, is that phospholipeptide ever going to be generated somewhere else? So if we give you a vaccine, is your big toe going to fall off? Or, are you going to get some sort of nasty brain inflammation or whatever? And, this is where I think, some of the human immunopeptidome project studies are really going to be valuable, where we can get these atlases, if you like, of what is the immunopeptidome of the brain, or this particular part of the brain, the pons or whatever it might be, and how often do we see A, this antigen being presented, and B, in the context of at least the common HLA molecules, are these peptides present or not? And the one thing that we do know, and I'll try and answer quickly, Catherine. I'm sure there'll be people that are ready to go to bed, particularly the Europeans. We do know that also when these PTMs are aberrant, this can often affect the way the antigen is processed, right? So you will get miscleavages, you will get redirected cleavage. And so you can generate, I guess, cryptic peptides in that context, which again will make them fairly cancer specific. So, I think the short answer is I think we should look at everything and see which is most efficacious.
Yeah, thank you. Thank you so much.
And Tony, if you don't mind, there's actually one more question here. It says, could you comment on immunopeptidomic analysis from non-classical or conditional APCs, example under inflammatory or oxidative stress?
Yeah, I mean, maybe Christina's better to answer that question than I am, but definitely you will see changes. The biggest change we've seen so far is the pro-inflammatory cytokines, and the clear explanation for that is induction of immunoproteasome in those particular cells. But you could think of all sorts of oxidative stress, ER stress, which will impact on antigen processing for sure. Hypoxia, you name it. I mean, you think about a tumor, for example, it's under a lot of different metabolic stresses. It can be whether there's appropriate angiogenesis or not will impact on whether it becomes more hypoxic and what's presented. If you irradiate it, you can change class 1 expression, and also potentially the types of antigens that are being available for presentation. So, I think the one thing that we're learning is that the immunopeptidome is quite plastic. And, at least with gamma treatment, you may only have 50% overlap between those two conditions in the peptides that are presented. So it looks quite different. And given that most of cancer immunotherapy is trying to induce inflammation in the tumor, it seems a little crazy that we aren't looking at both constitutively presented antigens and also antigens presented when there is infiltrating lymphocytes. So that's been the rationale for us to really look at tumor microenvironment and how that can impact on antigen presentation.
Great. Okay. Well, thank you, Tony, and thank you everyone again. I mean, Tony, that was a wonderful presentation. Andy, Christina, both your presentations were wonderful as well. And also thank you to our Peaks team for everyone who's put in their time and work into making this user meeting so wonderful with the research that we've shared today. I'm sorry it went much longer than anticipated, but it's really great to see like these interactions and interests within the Peaks community. As always, if there's any questions, comments, suggestions, please feel free to contact us by email or phone. And in addition, anyone who's attending ASMS next week, we do have a booth on the exhibitor floor. It's booth 315. If you walk straight through the entrance of the exhibitor area, we're right there. So otherwise, hopefully we'll be able to see everyone, all of you guys again soon in person. It has been a long time. But I mean, until then, wishing you all the best, and I hope you all stay safe and enjoy the rest of your day.
Thanks, everyone.
Thank you, Catherine. Thank you.
Thank you.
Thank you.
Thank you.